Good morning, everyone, and happy Monday. We're very happy to kick off session 4 of our CERSI course. Today, we're going to cover chemistry, manufacturing, and controls, typically abbreviated as CMC, for both drugs and biologics. Next slide, Nathan. And this flows nicely from the other 3 sessions that we've had already. So basically today, our instructors are going to describe CMC and quality considerations in the manufacture of drugs and biologics to support all phases of drug development. Next slide. So we are fortunate to have 2 great instructors today, Dr. Gayle Derfus and Dr. Eric Gorman, both of them from Gilead Sciences. Next slide. I will introduce them in a minute, but for now, just a couple of reminders. We having a session on Monday is unusual. Again, thank you for your flexibility, and I'm so glad most of you have been able to make it. We have a double session this week. On Thursday, we will have session 5 on biomarkers and companion diagnostics, and we are not going to have a session on Friday. Again, we have the slides from last week's session on toxicology posted in the website, so go take a look. As a reminder, please use the Slido for your Q&A. After discussing with the instructors, with Gayle and Eric today, we have decided that it would be best if instead of answering questions live as you post them, the speakers pause during the presentation and take dedicated Q&A time. Both Gayle and Eric have also agreed to stay until 1:30 to answer any additional questions. So the plan is to finish the session at around 1 p.m., and then they will stay behind for another half an hour. So for those of you that can stay from 1 to 1:30, that's another opportunity, another nice window to answer questions. Next slide. So I would like to start by introducing Dr. Gayle Derfus. Gayle is an executive director in drug substance process development at Gilead Sciences. Gayle has 15 years of experience in process development. She, at Gilead, she's responsible for end-to-end process development and manufacturing support for biologics drug substances. This includes things like cell line development, cell culture process development, purification process development, and pilot plant operations. And before that, Gayle worked at Genentech in upstream process development. Gayle has a PhD in bioengineering from UCSD and a bachelor's in engineering from Iowa State. Next slide. We also are fortunate to have Dr. Eric Gorman with us. Eric is Director of Formulation and Process Development, also at Gilead Sciences. Eric has 12 years of experience in preclinical drug candidate optimization, characterization of pre-formulation and formulation development, process scale-up, tech transfer, and validation of drug candidates. He was involved in the development of several marketed drugs, antivirals, Epclusa and Sunlenca, and also multiple clinical drug candidates. Eric has a PhD and a master's in pharmaceutical chemistry at the University of Kansas and a bachelor's in pharmaceutical sciences with a minor in chemistry at Drake University. So Gayle is going to get us started with biologic CMC. And I believe that, Gayle, you would like to share your screen.
Okay, thank you. Thank you for having me, and thank you for the nice introduction. So as Dolo said, I work at our biologics organization site in Oceanside. And so I'll be starting with a broad overview of some of the CMC activities with an emphasis on biologics, the way we do things for biologics, because that's what I know. And then Eric will take over and he'll do more of a case study on application of some of these CMC concepts, and then that's about small molecules. So you'll kind of get a flavor of both, where mine's a little bit more like a lecture and Eric's is more of a case study story. So our learning objectives for this part is to understand the role of the CMC teams in biologics drug development. And again, much of it applies to small molecule as well. Describe biologics and their manufacturing processes, and a little bit of understanding how they're different from small molecules. And then understand the concept of phase-appropriate product and process development and scale up. We really apply different strategies, whether if we're in early stage of development versus late stage trying to make a commercial product. And then I'll describe some challenges for biologics supply chain management as well. Okay, so where do we fit in to the life cycle of a product? So I think you've heard about a little bit about research already, who is the team that does the hardcore science to identify drug targets. And then once there's an idea for a drug, we get in to start doing some of the CMC activities or the development, where we try to define how are we going to make this drug. The clinical development group does studies to understand, does the drug work? And then eventually, if it does, you file the marketing application and you have a product that you manufacture and distribute to the market. And so the CMC teams are actually involved throughout this entire life cycle, the vast majority of it being during the development phase, but we also support research in identifying the drug targets and what type of molecule to choose, and we'll talk a little bit more about that. And then, of course, support marketed products as anything comes up that might require lab work or application filing support, things like that. So it is the CMC team's responsibility to make sure that we have a drug that is safe and effective, that it's consistent from what we put in the clinical trials all the way to what we put in the marketed product. And once it's a marketed product, that it's consistent across all the batches and lots that we produce. We have to make sure that we are continuously delivering the same product to the patients. So drug development is a long and rewarding road. I think you've probably heard a little bit about these phases of development and the filing applications already, but many compounds are considered in the preclinical or research phase of a project life cycle. And each one of those dots is meant to represent a compound. And after you file an IND, you can do a phase 1 study. So you'll have, quite a few molecules that you go in and try a phase 1 study. And then many of those don't make it to phase 2. And then some of those from phase 2 make it to phase 3. And eventually you have filing a marketing application and you get one product. And then what we're trying to show across the bottom is that a molecule could be in the preclinical stage for 2 to 5 years as they try to identify what is the right target for the mechanism of action that they're interested in, and also what's the right type of molecule. And then it can be anywhere from 3 would be really fast for a biologic, especially, to like 10 years from when you start your phase 1 study all the way to market. And then the blue is meant to represent, the dark blue is the cost. So as you go later and later into these stages of development, the cost increases in terms of the cost of investing in the manufacturing and the clinical studies themselves. And then these clinical studies have different purposes. So the first one, we're really trying to find out, is it safe? Can we figure out anything about the right dose? Sometimes figuring out the dose goes into phase 2 as well. But in phase 2, we're really trying to find out, does the drug work? Typically, there'll be more subjects involved in the phase 2 study. And then we'll go to a phase 3 if we're pretty sure it works and we need to demonstrate that. Well, it could be that it's better than the existing treatment. If there isn't an existing treatment, we show that it works in a statistically measurable way. So that'll be like a much larger study with many more subjects in it. And then across the bottom, we're showing that as you go from phase 1 to phase 2, because of the number of people in the studies keeps getting larger, you need to be making more and more drugs. So we'll talk about how we do that. And then you increase your product and process understanding as you go from phase 1 to phase 3. And this is partly what I was saying about the phase appropriate approach, where you invest more and more to learn more about your product and process as you get further along in the development. So we'll move on. So one of the first things that the CMC team gets involved with research on is defining a target product profile. So ideally, when you're developing a product, you know what you're trying to make from the start. So we know if we know what the indication is and the mechanism of action that we're trying to address, we can define the product and develop it to meet those requirements. And I just put an example in here of some of the CMC relevant elements of a TPP for HIV cure. This is a published paper, and the TPP includes many more things. Maybe some of them you've talked about in the other lectures, but I just put in a few here that would drive what we might do from a CMC perspective. So, for example, a target patient population, administration route, if like a parenteral injection is acceptable, or you need to have a single dose oral regimen, that really makes a difference in what we can do from the CMC side. The regimen duration determines how long our product has to be stable, and other things, how our manufacturing frequency gets set up. Storage is another thing we have to design around. So if cold storage is acceptable, we can we'll develop for that. If we have to have it stable at ambient temperature, we'll develop probably a different formulation or drug product presentation. So these are just examples of things that we, if we know up front, we can really design the product to meet those needs. And then we do contribute, this is, I think you've talked about the two main filings. So prior to starting the human studies is the IND, and then prior to marketing or selling the product is the NDA for small molecule, or BLA. And the main CMC information that goes in there is manufacturing process, and then also the product testing methods and results. So we're really demonstrating that we know how to make the product and that we can do it consistently, and then that we know what the product is, meaning we test many attributes about the product, and we'll talk about those in a little bit. And so we have to share how we do that testing and the results of the testing to show that the product is what we think it is. And then we also contribute when agencies ask questions. A lot of the times their questions will be that they want to understand more about the CMC aspects of the program as well. So we'll be involved in those anytime the agencies want to ask a question. Okay, so CMC teams are made up of many different functions, and this is a little bit biologics specific in terms of the naming of the functions, but I guess really it's just this first one, the drug substance. We develop, we'll develop the drug substance manufacturing process, and that's the bulk, we'll call it, where we put it into like a bag or a bottle. And then there's a team that develops drug product, which is taking that drug substance and filling it into vials or syringes or whatever the drug product configuration is that the patient will actually see. We have analytical development and quality control groups that develop those methods I was talking about. They generate the data to support that the manufacturing process is making what we say it should make. And they also support the regulatory strategy, especially when there are changes made to the process, and we'll talk about that. Quality assurance, make sure that we're adhering to all the quality standards for drug substance and drug product manufacturing and the testing. So all the documentation, they ensure all of our documentation is in place and accurate. And then regulatory compliance, they make sure that we are doing everything we need to make those filings successful. So they make sure that our strategy, again, for example, for a change of process, or if it's a new type of modality that we're working on, they really help make sure that we're collecting all the data that we need to have to go into those filings. And then the clinical supply management team makes sure that the drug gets labeled and stored correctly and shipped to the clinical sites on time. So they really work closely with the CMC team to get the drug product, and then with the clinical development or the teams that manage the clinical studies to make sure that the drug is getting there. So this is the high level structure of CMC teams. There are other ad hoc teams that can join at times, but these are the main ones. And then for small molecule, the drug substance is more typically called the API team. Okay, biologics. Okay, so defining biologics. One key feature that defines biologics and distinguishes them from small molecule counterparts is that they're made by living organisms, or sometimes they are living organisms themselves, whereas small molecules are made by chemical synthesis. And so we can talk about, in some ways, biologics are more complex to develop than small molecules, although I'm not saying small molecules are easy either. I've seen some of the small molecule chemistry processes, and it does not look straightforward to me either. But some of the ways that biologics are more complex is that the size of the molecule, we are not able to make a perfectly uniform product. It's typically a heterogeneous mixture. And so this is where the analytical testing comes in to really show that despite there being a mix of various product-related variants, that we're getting the same mix every single time we make it. And so that whereas a small molecule, you can more definitively confirm that you have the exact same single product every time. The production time for biologics is also quite a bit longer, just because of the time to scale up cells. They only grow as fast as they can grow, and then to culture them long enough to make the product. And then the manufacturing cost is also higher, mainly due to the cost of the raw materials to grow the cells and also to purify out the product from everything else that the cells made. These are just some examples. This is a little bit old now, it's 2019, but some examples of some biologics that you may have heard of. Also a couple of small molecules on here, but biologics are definitely have a share of the market. And there are lots of different modalities of biologics becoming marketed as well. So we'll talk about some of those in the next few slides. Okay, so these are some of the modalities. The most traditional or typical one is the monoclonal antibody, which is just like the antibodies that we have that our bodies automatically make, but we'll design it with the region that I'm highlighting. I don't know if you can see my arrow, but the top on this diagram, targeting a specific molecule or cell receptor in the body and then to have a desired action on that cell or molecule. We can also design similar molecules that we're showing here in two different colors because the two different sides bind two different molecules. So you might be able to bring two different cell types close together and have one cell kill the other one, or you might be able to, there are various different ways that you can use these to basically have two intended mechanism of action at the same time. And we have bifunctional, which is similar, but it's just an antibody with, for example, a peptide attached to one end that can draw a certain cell type near it, or it could be a cytokine that has a certain effect, and the antibody takes it to a place in the body where you want to have that effect. The Fc fusion we would use when we have a smaller peptide or molecule that we want to make a drug out of it, but it doesn't have a long circulation time in the body. So we'll add this so-called Fc portion of the antibody to the peptide to increase the circulation time in the body and enable the drug to have its effect longer. We have antibody drug conjugates, where the antibody is the backbone, and then there's a linker represented here as a black line, and a small molecule drug attached, which is represented by the stars. And you can have more than just four. I've just shown it as an example, but in this case, you use the antibody to take the drug to where you want it to go. And then when it gets there, the drug is released. And for example, if it's a cancer cell, the drug would be only there at the cancer cells and kill the cancer instead of going everywhere else in the body and releasing that drug. Sometimes these drugs are highly potent and you don't want them going anywhere besides the targeted cells. Some other non-protein modalities are viral vaccines and cell therapy, which is our Kite division manages the cell therapy treatments. They are live T cells that are engineered outside the body and returned to the body. So the modality or molecule type is selected typically by research based on the desired mechanism of action. And then the development or CMC team will start working on how to manufacture it. And so the things that we do to manufacture are mostly the same. If it's some of these recombinant protein formats on the left-hand side, the manufacturing process can be pretty similar for all of these. But if it's a viral vaccine or a T-cell therapy, the manufacturing process is very different. Any questions?
Yeah, can you go back to slide 12? There was a question regarding your table. So based on this, overall, it looks like a small molecule is more advantageous to a biologic. So why would you pick or market a biologic over a small molecule?
Yeah, because sometimes the small molecule can't do what we're trying to do with the biologic. So it's back to that mechanism of action, right? If we, like the ADC that I was just talking about, if we really need it to go to a certain site, or the other thing that the antibodies can do is elicit an immune response, so that that the small molecules can't do, right? So the antibody can go to a specific place, cause the immune system to go to that specific place and attack that cell line or whatever you're trying to do there. So it's really mainly about that mechanism of action, if it's something we need to try to do that a small molecule can't do. But yeah, and there are other potential downsides too, right? It's easy to make an oral tablet out of a small molecule. Biologics right now, we have to do an intravenous infusion or subcutaneous injection. So if we can get what we need from a small molecule, we would. Okay, so in the beginning of the CMC involvement with the research team, we do what we call a molecule assessment. So even within each modality type I was showing you, like even within just the standard antibodies, everyone is slightly different because the exact sequence is different, because they're targeting different locations in the body. So we have to do an assessment for potential liabilities, manufacturing related liabilities, meaning things that might make it hard to manufacture, or hopefully not impossible, but usually the research team will have maybe 4 to 6 different options for a given indication. They'll say, like, test out these 4 different antibodies or 6. And so we'll go through this assessment to see how well can we purify it. For example, for purification, one of the steps, I think we'll talk about that a little bit later, but one of the steps is to take the pH to a low level to inactivate any potential viruses. If we find out during this assessment that the antibody can't survive that or isn't stable, then we would recommend back to research, no, we can't have, we would choose a different one. The analytical team also identifies unique molecular features that maybe need to be monitored, or in some cases they have to develop new methods to be able to test and monitor those attributes. They also assess to make sure that the molecule is stable. And that's in collaboration with the drug product team, which is on the bottom left. So we do a quick assessment, maybe 4 weeks of stability testing, just put it in a potential formulation that we could use and make sure that the molecule is stable and doesn't degrade over time. If there's one that either degrades or various, for example, it could aggregate and the antibodies could all clump together. If we see that happening, we'll recommend choosing a different molecule there too. The cell line development team also establishes the methods that we're going to use to make the cell line. We'll talk about this a little bit more, but we kind of have standard methods that we can use. And then if it's a more complicated molecule, we might have to invest more to establish a different method that will take a little bit longer. So this early assessment is just kind of can we use our existing standard method, or are we going to have to invest more resources in order to choose this molecule? They also evaluate the expression level to make sure that we would be able to make the product at a reasonable quantity to serve the market. There are occasionally some molecules that really the cells just won't make it at a high enough level to make it a viable product. So that's our so-called molecule assessment or developability assessment in helping to choose what molecules actually come in for development. Okay, so now this is I was talking about how we can kind of use the same manufacturing process for all of the different recombinant proteins. And this is it at a very high level. So the first thing we do is we'll develop a cell line. So we use typically a Chinese hamster ovary cell line, transfect it with the DNA that encodes the protein sequence that we want to make. And that's about a 4-month process, believe it or not, to get to a cell line that's stable and expressing the product that we can, so we can use it in our manufacturing. And then the next step is to culture those cells in bioreactors. So that's what's represented in the pink container. And after the cells grow for a certain amount of time, make the product, it goes into a purification process. And this is meant to represent a column chromatography step. There are some other filtration steps we'll talk about as well. And the idea there is to remove everything except the protein of interest. And eventually we get to the end of the purification process and formulate it into what I was talking about earlier, the drug substance. So this will be 2 liter, 10 liter, even could be 100 liters, depending on the scale you're at, but a large bottle or bag full of the drug substance. So this is not what the patient will see, but it's a stopping point, an intermediate point where you can store it until you're ready to go to a drug product filling site, and you put the drug product gets put into vials or potentially syringes if it's going to be a subcutaneous delivery where we want it in a pre-filled syringe. So these are the kind of main manufacturing steps. And then all throughout this, the analytical testing is going on to make sure that the product is all the things I talked about earlier, safe, that it is what we think it is. There's checks along the way to make sure that we don't release any of this to the clinic unless it meets all the criteria. Okay, so a little bit more on cell line development. So the first thing we have to do once the research team has decided on the molecule they want to pursue is we prepare the DNA that will encode that molecule, combine it into a vector that then it also contains other elements to make sure that the protein will be expressed. We transfect that into the cell line. So now this vector, including the DNA for the product, gets integrated into the genome of the host cell. And like I said, this process is about 4 months by the time we go through all the steps to make sure that we have a single clone that we can use. So once this is done, we end up with just a big solution of cells. They plate it out into 96 well, like a microtiter plate, maybe you've heard of. And we measure how much product each clone is making. So each well, in theory, contains 1 clone. We do extra work throughout this process to make sure that we isolate a single clone so that we know that there's only 1 product being made by that, that there's only a single clone making the product, because every cell in our bank will have originated from that single clone. And then we'll typically test it in bioreactors and then measure the product quality to know that we're choosing a clone that makes the right product. And once we've chosen the final cell line, we will preserve a cell bank. And in the case of the cell line development lab, they're making what we call a pre-master cell bank or pre-MCB. And then we'll take that into a GMP environment and make the master cell bank, which is the critical raw material that defines our product. So this is what we start from every single time we want to make a product, is this cell bank. So that pre-MCB from the cell line development team, it's a vial, 1 milliliter of cells in media. You thaw it, expand it up in a culture until you have enough to make 200 to 500 1 milliliter vials. And then you freeze that down and store it in liquid nitrogen. We also test it to prove that it doesn't have any microbial contamination, that it is indeed making the product that we intended for it to make. And then it just gets stored in liquid nitrogen in the life of the product. And every time we want to make new product, we come back, thaw a vial of this. The thing you may be wondering how we don't run out. So once we get to the later stages of development, we'll make what we call a working cell bank from the master, where we thaw 1 vial, go through this process again and create another 2 to 500. and then we manufacture from the working cell bank. So we can always go back to this master cell bank and make more working cell banks so that we are always have a supply for our product. And then the
Gayle?
Yeah.
So we had a few more questions. They would just kind of interject here and just kind of catch up on those. So there was one I'll address really quickly that was around the average cost to make a small molecule for phase 1 trials.
Yeah.
It's difficult to say because it's going to depend on cost for raw materials and the complexity of synthesis. But you're probably looking at on the order of $1 million or so, but it's going to depend on the molecule for like phase 1 supply for a small molecule. Couple for you, Gayle. So one was, what's the difference between cell line development and cell culture? Isn't cells growing during both of the processes?
Yes, very good observation. So during cell line development, absolutely, we are growing the cells in culture. So it's kind of just the nomenclature that we use. And we'll get to what I'm talking about with cell culture, but absolutely the cells are being cultured to generate the cell line as well. So it's really just the way we divide our work more than I did not mean to imply that the cells are not being cultured during cell line development. Yeah.
And then another was about the cell lines. So why that particular cell line? So hamster ovary cells, usually used to create the cell line that produces the product during manufacturing.
Yeah. So why that as opposed to, say, a bacterial organism is because they can do what we call the post-translational modifications. So glycosylation is a big one for these types of proteins. Though the mammalian cells can do that, whereas microbial cell lines cannot. And why CHO among the mammalian cell lines is really because it's the industrial standard and it's very well understood across the industry, accepted by the agencies. They all are familiar with it. If you say you started with CHO, they know what you're talking about. They know that it's well understood. You know, the things that you need to think about if you're using that cell line. It's not to say you cannot use a different one. There are manufacturing processes that use other murine cell lines, for example, and there are commercially marketed products that don't use CHO. So it's not to say you have to, it's just a more typical.
And we just got one new one, but I'll ask while you're talking. So how do you pick a stable cell line? So how do you pick the best one from the different transfected clones?
Yeah, I really glossed over that quickly. So best at first, so there are actually I skipped a few rounds in here between evaluate candidate clones and select final clones. So these are 96, well, forget what fits in there, 300 microliters, right? In the beginning, best just means the ones that are making the product at all. I think the white dots there are meant to represent one that's not even making any, right? So in the beginning, we're just trying to find the clones that are making it. We gradually culture them in larger vessel sizes. And when we get to the point of 15 milliliter cultures, we actually measure the product quality. And we'll talk about some of those aspects of product quality that we look for. But at that point, we won't choose one, for example, that is aggregated where the proteins are all stuck together, or one that is it's rare that one would be falling apart. And you can tell that on a SDS page kind of measurement. But so it's at the beginning, it's how much they're making. And at the end, it's how much they're making plus is a good product. A couple other considerations is, say we check all those boxes with our top, 6 clones or something. So they're all good. Then we'll try to go with one that grows similarly to our previous, in terms of how high the cell density gets. We'll go with one that's similar in growth to our previous products so that we know our manufacturing process will be easy to develop, meaning it should be similar to ones that we've done in the past. But mainly it's the productivity and product quality. Anything else? Okay.
That's it for now.
Okay, so what is cell culture? So this kind of will help answer the other question too. And again, it's really just how we divide the work. But once the cell bank is made, then we go into what we call the cell culture process for making the drug. So we start from, again, 1 milliliter of cells. You have to expand the culture So each day, so it starts at, we take that 1 milliliter, put it into 30 milliliters of media. They grow for 3 days, then you're at a high cell density, and we do what we call passage, where we take that 30 milliliters. This is meant to be a bigger one, and so say that one goes up to 250 milliliters. You take that full 30 milliliters, dilute it up to 250 in media, and then they grow for 3 days, and then they're ready to passage again. And we'll start in vessels like shake flasks that are in an incubator. So it's just CO2, like carbon dioxide and temperature control. And then eventually we switch into our bioreactors and just keep expanding the culture volume as much as we need to until we have enough cells to inoculate what we call the production bioreactor. And so then that production bioreactor, we let the cells grow for 14 to 16 days. And so at this point, we aren't going to passage anymore. We let them grow, they grow to high density, and eventually they start making the product. So very little product is actually made during these expansion stages. And we don't even harvest any of it. We just keep getting more and more cells until we can run this production bioreactor. And so at the end of that culture, we have the cells still, we have all the media components that the cells didn't use. We have other byproducts that they made, and I guess that's about it. So that is not the drug at the end of cell culture. But yeah, so this is the so-called cell culture step of manufacturing. Any questions on that one?
There was a question, how do you check the cell viability after thawing the cells on slide 18? And 2 more just came in.
On slide 18, so when we thaw them, we have cell counters, just like any, you can do it manually. We have automated ones here where it mixes the cell culture. You just take like 1 milliliter or less out of the culture, mix it with trypan blue, and that dye gets into inside the cell if the cell is dead, and it stays out if it's alive. So then it goes through imaging process that counts the live cells that aren't stained, and it counts the ones that are, computes the viability. So it's actually quite nice if you come from an academic lab and come work in a lab in the industry. Automated, it's nice. And that's the same thing actually that we do throughout. So every time we take a sample, we're monitoring these cultures all the way through. So we inoculate it on day 0, take a sample, measure how many cells are in there and the viability. And we keep doing that all the way throughout. We measure the viability from beginning to end of this production bioreactor as well.
So another question was, how do you assess stability of the cell line during scale up? So the ability to produce the product.
Yeah, so that's another step that I skipped in the cell line selection slide. So once we have, say we have 4 or 6 clones that make a good product, we want to choose one of them. We will grow them for 60 days. It doesn't actually show that we need 60 days here, but if we get to larger scale manufacturing, sometimes this expansion stage is longer. So we'll cover up to 60-ish days and do experiments, production cultures along the way to show that even after staying in culture for 60 days, they still produce enough product and that the product quality has not changed. So that's another checkbox in choosing the cell line that I just didn't mention it earlier. But we would know that before we go into the actual manufacturing.
And then the last was, what sort of volumes are typically used for bioreactor stage? But I think you're covering that on an upcoming slide.
Yes, we will get to that. Can't remember how many more slides from now, but we will. Okay, so then the recovery and purification. So harvest is the next step right after the bioreactor. That can be done with a centrifuge, like a giant centrifuge that can take up a whole room, or a centrifuge combined with what we call a depth filter. But the main point of that step is to remove the cells and other debris, but keeping the product. But at this point, a lot of the other things I mentioned that are still in there have not been removed. So then we have 3 different chromatography steps. We have a protein A, and then typically I guess I only showed one on here, but sometimes there will be 2 other, like a cation exchange or anion exchange chromatography step. And those are really to remove any impurities from the process, the host cells are making other proteins. We call them host cell proteins or HCP. The DNA from the cells is still sometimes it's still in there. It doesn't get removed during the harvest. And the other cell culture media components are removed. Sometimes if there's a aggregated mAb, for example, that can be removed through these steps as well. The other main thing that the downstream or the purification manufacturing needs to do is show that if there were viruses introduced at any point prior to that, that we could get rid of them. So we're not expecting any to be in there. There aren't any intentionally introduced, but there's the low pH fold that I mentioned earlier when I was talking about molecule selection. So after the protein A chromatography, we'll take it to low pH. That would inactivate certain viruses, and we'll do studies to show that, to prove that if they were there, well, you spike them in a small scale study in material that's not going into any animal or human, and show that low pH fold inactivates certain viruses. You also can show that the polishing chromatography steps can remove viruses. So you'll show how much you can reduce viral load from there. And then there's a virus filtration step that should remove any other. So we have 3 to 4 different steps where we can show that we would remove any viruses that could have been introduced. And then you get to this ultrafiltration, diafiltration step where you concentrate the protein to the concentration that's going to be needed for the clinic or for the product, and exchange it into the final formulation. So this is essentially generating the drug substance that will be stored in those bags or bottles that I talked about. And I think the next one is about, so how we choose that formulation. Well, any questions before I move to this one?
There was one, but I think you're going to cover it upcoming about testing activity, showing functionality. There was one, what is involved with a low pH hold? Does it change or denature the drug product given it's a protein?
Right. So it doesn't, because we choose. So in that early molecule assessment or developability assessment that I was talking about, we make sure that we find a protein that won't be denatured or destroyed by that low pH hold. But it's really just titrate the pH down to a low pH, like 3, and hold it there for a certain amount of time. And then we bring it back up to finish the processing. It's pretty simple. Okay, and then we'll get to some of the testing eventually here too.
Another one just came in. Why are there so many steps for detecting viral infection? Is bacterial infection not a concern?
So we do a lot of things to protect from bacterial infection as well. So that cell line, when I talked about the cell line development, at the end of that, you test it to show that it does not contain any bacterial contamination. We also filter all of our buffers that we use. So the solutions that we use to do this purification, the media before we grow the cells in it, we sterilize all the culture vessels before we use them to make sure that, again, no bacterial contaminations are introduced. It's pretty obvious if you get a bacterial contamination because you see it and it's a mess and you have to toss the whole batch. So viruses, on the other hand, are a little bit harder. So you really have to focus on this theoretical showing that you would eliminate it if it was there. Also for the bacterial, we do testing at all the steps along the way. And it's more on the analytical side, but they test for bioburden and endotoxin, which would indicate that if there had been a bacterial contamination, and say you filtered out the bacteria already, but they left any endotoxins in there, we test for that. And so if it's over a certain very, very low limit, we can't use the batch. So we are definitely protecting for that as well. Okay. These are good questions. Okay, so the formulation team or drug product development team, they prepare for all of the steps in the process to get from that stored drug product that I was talking about to the patient. And the first thing they do is choose the formulation. So not every protein is stable in the same formulation. So what sugar we put in there. Sometimes it needs stabilizers. So they'll study in a range of also what pH to store it at. So they'll do studies to look at all the different possibilities and show which one is the most stable and choose that. And so the drug product team then communicates that to the drug substance team, and the drug substance team does that final UFDF step that I was talking about here and into that final formulation that the drug product team has identified. They also have to decide if it's going to be, if it is stable as a liquid, that's what we would choose as our primary option. If it's not, we have to do what we call lyophilize it, which is freeze dry, essentially. So then it comes out as a drug product that is a white cake that then has to be reconstituted. The drug product team also manages the site that's doing the fill itself. So there's some aspects of that manufacturing process that need to be controlled as well. So if it's a lyophilized product, when it's time to actually use it, you have to reconstitute it in that vial with water, transfer it to a syringe, and then from that syringe, inject it into an IV bag that has saline or dextrose, whatever the indicated solution is, and then it goes to the patient from the IV bag. If it's not an IV, if it's a subcutaneous dosing, of course they can just stop at the syringe step and it can be injected from there. There's also a pharmacy manual that is provided to the pharmacy to make sure that they do this reconstitution correctly, that they inject the right amount into the IV bag, or that they have the right amount in the syringe. Another thing that we have to do for drug product is make sure that like I said, there's a study to make sure it's stable in the vial. We have to do studies to show it's stable in a syringe or an IV bag as well. The vials we intend to store years, like 3 years, 5 years, if we can. Whereas the studies to support using the IV bag and the syringe are more on the order of a day-ish, right? Because you're just going to put it in there and give it to the patient. So all the data has to be collected to support that the product is sterile and safe and does not change throughout this entire process. Any questions on this one?
Gayle, so I think going back, so there was a question that came in around, how about mycoplasma contamination? Is it tested every step during scale-up?
Not every step, no. So it's tested on the cell bank to make sure that right from the start, we don't have any, right? And then we do a test at the end of the production bioreactor to make sure that there's nothing there.
So another fun one. So mentioned the product is held in plastic containers until transferred into glass vials for formulations and other things. Why keep it in plastic rather than glass? Isn't plastic more reactive and likely to transfer unwanted chemicals into the final product?
So that's a good question. So we do studies to make sure that the plastic does not do that. That we know any possible thing, any possible chemical or molecule that can be transferred into the product. I don't actually know what happens if you freeze a big glass container. But so that's a good question though. I don't actually know that it would be impossible to use glass for that, but we do store it frozen, and I think that might not work so well for a glass container.
And then another one just came in. Compatibility study with only the IV bag is sufficient, or do you also need to be performed with a syringe?
Yeah. So whatever the entire process is going to be in the clinic or at point of use, we do the compatibility study for the whole entire process. So we reproduce the whole thing as it will be done, except that if it's going through a syringe, we store the syringe for 4 hours, 6 hours, whatever it is, to do the max possible time that we're going to say it can be in the syringe, show that that's okay, then put it in the IV bag. So we do the all parts of what the drug will see on the way to the patient.
Maybe just for everybody's awareness, we're not going to talk about it in detail, but these are all covered under extractables and leachables is kind of the testing and characterization that are used. So you can find lots of guidance from the FDA and other agencies around this as well.
Yeah, and we'll choose the IV bag and syringe and vials that are well established as to what the extractables, leachables would be. Same thing for the drug substance storage at this point. But yes, we do that assessment for everything that's used.
And filters are also in scope as well, any filtering that's done as well.
Yeah. Yes, absolutely.
Okay, I think we're good to continue.
Okay, so a little bit more on how they choose the formulation. Or I guess it's the formulation and the configuration. So if we're going IV, we typically don't need super high concentration drug product, meaning that you have that 500 ml IV bag anyway. It doesn't really matter if you're putting 3 milliliters of drug in it or 30. But if you're trying to do a subcutaneous delivery, you can only fit maybe 2 to 3 milliliters under the skin, and that's even a lot. So in that case, you really want to have high concentration drug. So if we know that based on the TPP going in, that the goal is subcutaneous delivery, we will develop a formulation that is high concentration so that that's even feasible. So that's how we choose the drug concentration. The excipients are those other components I was talking about as buffers, stabilizers. We choose excipients, meaning the source of the excipients is critical. So we make sure that they are GMP grade, that they've been fully tested. There's no additional purification after we do that final formulation step. So everything has to come in sterile and high grade excipients. We use, FDA has a GRAS or GRAS designation of certain excipients. So we stick to those at all, wherever possible, just so that we know that they are approved excipients for this purpose. We talked about with choosing between liquid and lyophilized, the storage temperature. Some proteins are stable at room temperature, or not room, sorry, at 2 to 8 degrees, like refrigerated, and some are not. In that case, we need to freeze dry. The vial size and fill volume also depend on how much we are planning to dose. So if it's a high dose, we'll make a larger vial with a larger amount so that to make it easier on the clinician to not have to open up 10 vials and do 10 transfers into the IV bag. And then of course, if a device is needed, like a syringe or a pump or anything like that, we'll work on that. We'll start designing that to go with it as well. Okay.
Yeah, just to address a question that came up just for everybody. There was a question around after the last step of purification, will the entire batch be lyophilized? So just to make sure it's clear, the lyophilization happens for the final container, not an intermediate. So it wouldn't be the bulk material, it would be the final product in the vial that would be lyophilized, but it would generally be the entire batch together. And then volumes that you would see at that step, that'll be discussed in about 10, 15 more slides.
Okay. So then the actual manufacturing of those vials is done aseptically, meaning under sterile conditions. So we thaw the drug substance in whatever container that it's in. You can pool and mix the drug substance batches. So if you need like 2 bottles or 2 bags to make the number of vials you want to make, you can do that in that step. You can also dilute it if your drug substance is at a higher concentration than what you want your drug product vials to be filled at. You could dilute in the same solution that the protein is already formulated in. And then we do filtration for bioburden reduction, make sure it's sterile. And then after that, do a filter integrity test to prove that the filter did what it was supposed to do. It was not compromised in any way. And then the vials are filled in an automated filling line. After they're filled, there's a weight check to make sure that they were filled to the right volume that they needed to be filled to. They get a stopper and a cap put on and an overseal. And then they're visually inspected and packaged up and then at that point, we call them bright stock. They're not actually labeled because depending on where they're going into what country we're doing a study in or what study we're doing, sometimes the label needs to contain different information. So at this point, they're just vials and the outer packaging will tell what it is. And then just so no one worries that we might mix it up, we do ID testing, which we'll talk about in a minute, once we're going to do the labeling, we'll do ID testing to prove that what we shipped is that we know what was in that vial, essentially. Okay.
Gayle,
Yeah.
can you clarify what does bioburden reduction mean here?
So it's another one of those, like, if there had been any potential bacterial contamination, that it would remove it. So it's a low pore size filter that would remove any potential. Again, we don't think that there's going to be any, but it's a precautionary measure to make sure that if there was any microbial, that we would be removing it before we put the drug into the vials.
And just to clarify too, that it's not isolated to biologics. That is true of any kind of sterile products with small molecules as well, if it's intended for like parenteral administration.
Okay. So then the supply chain team that I was mentioning earlier, they will do the labeling and packing, labeling, and distribution. So they work really closely with the clinicians that are doing the study to make sure that we get it labeled properly, that it's safely transported, and on time for the dosing. So they work to plan the demand based on information from the clinical study design. They choose what we call a depot, which is a storage site for the drug product vials, to be somewhere near where the studies will be. And then they when the study is going to start up, they label the study, manage the site that's doing the label. Sometimes that's not an internal site, and so there's work there to manage that. And then the secondary packaging site will put all the labeled vials into another packaging to enable shipping. And then this team also manages the study, the inventory for the study. So they keep track of how much each clinical site has used, continue distributing more as needed, and then also communicate back to the drug substance and drug product teams. Oh, we're running low, we're going to need to make more. So that resupply planning is a critical point in making sure that we don't short the clinic or eventually, hopefully, the market. And then in a study closeout, they'll make sure that all the inventory that wasn't used, that we know what happens to it. It either gets returned or documented that it's disposed of properly to make sure that every vial is accounted for. Okay, so finally, the testing question. So what are we looking for with this QC? We call it quality control testing or analytical testing. One is the identity. We have to show that it is the product that we intended to manufacture. We also look for what we call product-related species and product-related impurities. So the product-related species are molecular variants that have comparable potency and immunogenicity. So it's okay that they're there. We just want to make sure that there's the same amount of each one every time. The impurities are anything that might have a different potency or immunogenicity. In that case, we make sure that we're, again, having them at low level or removing them and demonstrating that with the testing. We also have to test the potency. So that is like how strong the drug is, like how well does it work for the mechanism of action that it's intended to have. And that's a key element that we need to make sure that is consistent for every batch that we make to be able to release it. And then we also have process impurities. So we talked about some of these that they come from the manufacturing process. So the other things that the cells, other proteins that the cells make, DNA, the media components. So there are a lot of different nutrients that we provide the cells that we need to get rid of that are not going to be part of the product. And then even the purification process itself can introduce impurities. So that first step that I was talking about, the protein A column chromatography, we have to show that we don't have any, by the end of the process, we don't have any leached protein A still in the drug product. So we have testing for that as well. And so these are kind of high level look at what the tests are that we do, but each one of these, especially the product related species and impurities, are quite a few different tests that we actually run. I don't know, did I answer the question about the testing, or is there more? I forgot what.
I think so. Mostly it was around making sure that you're actually testing like potency and things. So I think it was addressed.
Yeah. Yep. And we test along the way. So we test the drug substance for many of these things, and we also test the drug product. So there, you know, any chance that anything changed, there shouldn't be, right? Because the drug product manufacturing process is pretty, like, you don't actually change the product, but we test anyway to make sure. We have to be absolutely sure that we're meeting all the criteria that we set ahead of time for the product. Yeah. Okay. Okay, so any questions before we launch into phase appropriate development strategies? Okay. So we kind of divide into early and late stage for biologics development. In the early stage, that's like before we submit the IND to do the study and through the phase 2 trials. The focus is speed to clinic. So you saw all the potential molecules, many of them fail during phase 1 or 2. So the idea is to get as many of them as possible to phase 1 and 2 quickly and find out, do they work or not? Just find out. And then if something is if a molecule will make it, then we switch over to late stage development and do what we call molecule specific development. So in early stage, we'll use what we call a platform process, meaning standard manufacturing processes. So we don't really tailor it very specifically to each molecule if we don't have to. Same with the test methods. Sometimes we have to, but if we can get by with the ones we have and produce still the product that we need and it's safe, then we go with that. But in the late stage, we will optimize the process specifically for that molecule to try to get as high of productivity as we can, and also make sure that it's a really robust process, meaning we're not operating on the edge of, if the pH changed by a slight amount, then the product quality goes off or something like that. So we really do what we call the DOE, or design of experiment studies, to make sure that we really understand how our process is running, and are we at a place where there can be some variability in process and still produce a high quality product. And so at that point, you're making for the phase 3, which is typically pivotal. Sometimes a phase 2 study can be pivotal as well, but pivotal is the one that you're going to submit the data to the FDA to register your product. And then we try to use that same process for pivotal studies as what we intend to use for the commercial supply. So sometimes we will, because we are optimizing for the late stage, there will be process changes between what we use to make the early stage product and what we use in late stage. And I think I said this already, but it's typically if we need to increase the yield, like we're not making very much because we went with the standard process. If we change a few things here and there, we can make more per batch. But anytime we make these changes, we always have to make sure that the product is comparable, and that's at minimum, I think I have a slide about this too, but at minimum, an analytical testing assessment to show that these changes did not change what the actual product is. Manufacturing changes didn't change the product. We also might change methods. I said we start with standard test methods. We can use more product specific methods, and we're expected to, in fact, for potency, to develop a potency assay that's very reflective of the mechanism of action for the later stages. So as we understand the product more, we might tweak the assays to make sure we're measuring all the very important aspects of the product as well. So to sum it up, a lot more investment in the manufacturing and testing in later stage, because at that point you really need it to be a robust supply for eventually serving the market.
Gayle, a couple more questions came in around the QC testing, and a couple other ones just kind of around this slide. So regarding the testing, how is potency measured during QC? Is it the same binding assays used in discovery?
So yeah, so in the early stage, we can use like an ELISA, but later stage, it typically needs to be a cell-based assay, which they wouldn't develop for research, right? Because it's a lot of work to develop that assay, and we don't even need to have it early stage either. So later on, yeah, the potency assay becomes typically a cell-based assay that's even more representative of the mechanism of action. What else?
Okay. Is potency only relevant for liquid formulations? So in other words, would a lyophilized product be pure or be effectively 100% potency?
No, so we to do the potency assay for a lyophilized product, we would reconstitute it and then do the same type of potency assay. So it's absolutely still relevant.
So then more towards this clinical information. Does this mean that you can change the formulation between early and late phases of development or between clinical trials?
Yeah, you can, but you have to do all the homework to show that that does not impact safety, efficacy, anything about the product, right? So you can do it. It's easier if you don't have to, but.
And then the last one, are there specific process changes that are not accepted when moving from a completed phase 1/2 to a phase 3 study? Is there a situation where the study needs to be repeated for safety in a small cohort prior to phase 3 because of product change?
Yeah, so I'm trying to think, I can't think of anything that's like, well, I don't know, maybe like you can even change the cell line that's making the same thing. I mean, you couldn't probably make drastic changes to the actual like amino acid sequence and still call it the same product. But at the end of the day, it's really all about that comparability package, which yes, the first step is the analytical comparability from testing. And then you may also have to do what the person mentioned, which is another safety study, or yeah, hopefully you don't have to go all the way back to the beginning, right? But depending on what the analytical comparability looks like, you may have to do those other studies as well.
And then another one just came in, and this might be addressed on a subsequent slide, so we'll see. It's often said that products fail at the scaling up step. Can you talk a little bit about what the risky steps are during the entire CMC process?
There are a lot. So fail at scale up. So yeah, so the cell culture process is probably the most likely part where you get a surprise at scale compared to the downstream or the purification. Sometimes it's this not really the scale itself, but more as you are scaling up and starting to make more and more batches, you start to see raw material variability of the incoming ingredients for the process. I think we are going to talk about some of the scale up challenges as we go on in this lecture. I'm trying to think yeah, it's probably just the cell culture, the production bioreactor. You can have like slightly different, despite we'll talk about some of the methods for scaling up, but despite matching the things you can match, sometimes you still get slightly different performance or maybe local gradients within the bioreactor, things like that you can't control. Yeah.
And then we just got one more new one. Are there any in silico tools based on molecule-specific features that can be used to predict potential purification problems or other CMC-related issues?
Yeah, they do that. So that you can predict a lot of potential spots for degradation of instability. So some of those we just know, and we'll search for those and make sure that like we don't choose a sequence that has those in there. Yeah, that's definitely used. Okay.
I think we're good.
Okay, so this one is these slides that are coming now are kind of wordy. We don't need to talk through every single thing in here, but we're like each one is for one of the different manufacturing steps. In early stage for cell culture, we talked about developing the cell line and making the cell bank. And as we proceed, we optimize the process if we need to, as I talked about, create working cell banks. I mentioned that. Potentially transfer to another site or a commercial contract manufacturing organization if the sponsor company doesn't have large scale manufacturing. We define scale down models to do all the studies to show that the process is robust. Eventually, we do what we call process performance qualification, which is sometimes just called validation batches. So you'll do at your final commercial scale that you intend to market from, you'll do 3 or 4 batches in sequence to show that your process is under control, that it meets all of the specifications that you set. We'll do more genetic characterization of the cell line and show that again, that it's stable all the way throughout as long as we would need it. We'll show it at the manufacturing scale, whereas early stage, it's shown at the small scale. And then write a lot of reports, make sure everything is documented really well so that you could go into filing. And then after you file the BLA, there's still lifecycle management. If you have to update anything about the process, of course, it's another comparability exercise and filing to enable that. And then we do what we call continued process verification. So we decide on certain performance metrics for the process and monitor them for every batch, and just we trend those to make sure that nothing's changing over time. For purification, it's a lot of the same things. They established that virus clearance capability in the beginning. I think most of the things are similar throughout development, but conduct resin reuse studies to show how many times you can reuse the resins in those columns before they don't work anymore. I think a lot of this is the same. Yeah, they also have to, purification also has to do scale down model development to show that the purification process is robust. And then the formulation and drug product, we talked about how they do stability studies to support formulation selection. They also establish the shelf life based on these studies. So we can only use the drug product for so long after it's made, and we have to define that based on studies. Later on in development, we'll decide if a formulation change is needed, whether the standard or platform one works. If it does, it's great. If it needs to be changed for whatever reason, we would do that. We also go into a little more detail in characterizing the drug product itself. And I think Eric mentioned the extractables and leachables. That study happens as you get farther into development. They do what we call primary stability studies. So as soon as you know the manufacturing site that you'll use, a site and scale and process and everything that you'll use for your commercial product, you have to run that way and set up stability studies so that you can claim a certain shelf life at the time of filing. So the drug product team works on that as well. Sometimes drug product scale up is needed as well. If you're making small number of vials per batch for clinical, maybe you need to scale up to larger number of vials per batch for commercial. Okay, and for analytical, we already talked about the molecule liabilities. One thing they do in preclinical is set up a reference standard. So they'll, from an early batch, create vials of product that then are used in the assays subsequently to show, a lot of times the assay result is it comparable to reference standard or not? So that starts in the early stage. They qualify assays, define the specifications to be able to test and release the drug substance and drug product. That starts all the way from preclinical. The development group transfers methods to QC labs to do that testing. And then as the development goes on, we identify a lot more, we develop a lot more understanding of the product. So there's this concept of critical quality attributes. We have to define what matters about the product in terms of the quality attributes that we measure, what matters for safety or efficacy or any other aspect of the way that the drug is working. We have to understand that, and so the analytical team is doing those studies during the development. Let's see what else. They're identifying any variants that impact degradation pathways, so we have to make sure that the tests that we're using to test on these stability studies, the primary stability studies that I talked about, for example, that the tests that we're doing on those over at each time point are actually representing any potential degradation that could happen. So that we call it stability indicating. We have to make sure that the test actually would show it if the product wasn't stable. Specifications can be revised. Most typically they get narrowed as you get more manufacturing experience. You can start early stage with somewhat wide because you don't have a lot of experience, but as you make more and more batches or lots, you understand what your manufacturing process is capable of. And so typically you're tightening the specifications. And then, yeah, okay, I think I'll let you guys read those later. So then, okay, any questions?
So we had one going back to, so for genetic characterization,
Mhm.
do you usually use next generation sequencing?
Yes, and yes, and sorry, I'm blanking on the other, but we also, yes, so we do that, and we also take the product itself and characterize it, like the protein itself, yeah. In the later stage, when we're, so the one that I was showing on the slide is like the at scale. So yeah, we'll age the cells, grow them through the final, and use them in the final manufacturing scale, and then yeah, do the next gen sequencing and also take the product and analyze it.
Okay. And then a few more kind of around the actual like PSBs and analytical testing. So regarding primary stability batches, if there are process changes afterwards, how does that affect primary stability?
Set it up again. Like start.
Basically, any change composition process, start it again.
So you could potentially like leverage still some of the stability information from before the change, right? But eventually you need to like get the actual manufacturing process stability data.
Another one, does primary stability for biologics need to be from batches at the same scale as intended commercial scale?
Yes.
And then what studies are needed to ensure product quality if product is being transferred from one CMO to another?
Sorry, what studies are needed to show comparability?
Basic comparability when you're doing tech transfers from one site to another.
Yeah, so we like typically would compare the release test results. Actually, that's here on the slide. So the release testing results will compare head to head, right? If it's early stage, you typically might just have like 1 or 2 batches of the transferring site and the new site. So it's not like you can really do statistics on it, right? But we'll compare them head to head. We also have what we call extended characterization, which these are test methods that are not on the release specification, right? So we don't do them in order to release the product for use, but it's additional information about the product that we'll do. For example, characterize the glycosylation patterns. That's one. So we'll go through those methods for both and show that the product is similar by that assessment. And then we also compare the stability of both. So you can, well, the stability at the intended storage condition, the stability at an accelerated or higher temperature, and then stability at a stress condition, right? Because the stability at the intended storage condition might show nothing, right? And so what if they're both, of course, they're both stable because that's how you're going to store it, but to really show it's the same product, ideally they have the same degradation pathway at a stress condition as well.
That was it for now.
Okay, yeah, so that's essentially what this slide was about, is if you make a change to cell line, you change the process or like the culture media, or you change one of the downstream steps, or you change the manufacturing site, you change the manufacturing scale, all kinds of different changes that can be made. And you do sort of a risk-based approach to determine the extent of the exercise. Also, if it's later in development, you'll have more batches at each pre and post change, so you can do more of a statistical assessment of these things as well. And then if, it may also be determined that the change is big enough that you do need to do the non-clinical safety or even another clinical study with after the change. So it's all, risk-based. Okay, scale up. I think we had a question about this a ways back. I think the next slide might actually answer the question, but so why scale up? Mainly to get more product per batch. There are 2 ways to get more product per batch. Well, 2 strategies, let's say. You can improve the process to get more protein per liter. We call the grams per liter is the titer. You can somewhat improve by increasing the yield of the downstream. So each step you recover a certain percent of the protein. That can have some impact on improving the per batch yield as well. The other strategy is to scale up. So if you're manufacturing at the 2,000 liter scale, you can switch to 10,000 liter scale, and typically the cost of the batch doesn't go up as quickly as the volume of the bioreactor goes up. So you get more product per cost if you scale up. Also, the number of batches required per year is a lot lower if you have the higher productivity process, but also, of course, if you're making 10,000 liter batches, you get a lot more product than you would for getting 2,000 liter batches. And sometimes it's a facility constraint, right? If you're at a facility that can only do 50 batches per year at the 2K scale, you might not be able to even supply your study or your market with the capacity of the facility, so you'll need to scale up. Oh, this one I think is just kind of what I was talking about in a somewhat graphical form. So titer or productivity is on the x-axis. As you increase titer, cost per gram goes down. Increasing the yield of the downstream purification process can slightly decrease the cost, but not quite. There's not a lot of leverage there. And then increasing the scale really does dramatically decrease the cost per gram, especially if your titer is low. Okay, this was the question that somebody asked. So these are just example scales or typical scales. The actual scale that you're going to use depends on early stage, like how much material you need for your studies, and then at commercial, of course, the market demand. But what I'm showing here is that like vial thaw all the way through the expansion to the production scale being the final bioreactor that you're seeing. So we have what we call pilot plants that run, they're not GMP, but they're somewhere in between GMP and what we do in the labs. And they make the material for animal studies. So this is typically like 100 liter, 250, 500 liters, somewhere on that order of magnitude. And that's typically enough for not only the animal studies, but also generating material for those formulation development studies I was talking about, and even purification process development. They'll use material like drug from this to develop the processes and formulations as well. And then for clinical scale, for phase 1 or 2, typically 2,000 liter is sufficient. And then even phase 3, sometimes you can get away with 2,000 liter, but ideally you would do your phase 3 or pivotal study in the same scale that you're planning to commercialize, which depends on your demand. You'll choose the size for commercial based on anticipated demand. So that for many products is 10,000 liter, 15,000 liter, 20,000 liter. But for some products, the demand is low or the productivity is so high that you can do your commercial process at the 2,000 liter scale and be just fine. So this part really depends on the process and the demand, what scale you choose for commercial. Did I answer the question about that one, or is there any more?
I think it got covered.
Okay.
We had another one come in. So it seems preferable to minimize the number of drug substance batches. Is there any industrial kind of general preference regarding how many drug product batches can be produced from one drug substance batch?
Oh, from drug substance to drug product, not really. Of course, like for traceability and, in potential future investigations, it's really simple. If it's one drug substance batch goes to one drug product batch, but it really doesn't have to be that. I think what I was saying too, for drug substance, minimizing number of batches helps in some sense once it's commercial, right? Because then you have the overhead of all the testing per batch. If it's 10,000 liters, you have to do it once. If it's like 5 by 2,000 liters, you have to do it 5 times. And all the documentation and everything that goes with it. That's a reason to minimize the number of drug substance batches per year, let's say, right? Early stage or like development, like clinical stage before it's commercial, sometimes you want to have more batches, right? Because like we were talking about for the comparability, like if you just have one batch and you did all your studies with that and then you make a change, you kind of don't have a lot of you have no variability in what's been in the clinic so far, first of all, and you also can't really, it's hard to assess comparability for N=1. So sometimes it's better if you do more batches in the clinical phases to have more experience of how understanding the variability of your manufacturing process as well. So long answer to that one.
I think the thing I'd add, Gayle, is also you don't want your drug substance to be around for too long. So you don't want to be just targeting only making drug substance every 2, 3, 4, 5 years, because then you've got to support the stability of that drug substance, but also you have more difficulty in adapting to changes in demands for the clinic or for commercial products.
Yeah, good point. Yeah. Yep.
I think that's it for now.
Yeah. Okay. Some scale-up considerations for cell culture. We choose, this maybe goes back to that question of some of the challenges, but we choose the operating conditions to try to get consistent performance at all the scales. Some things are directly controlled by the bioreactor control system, like temperature, pH, dissolved oxygen, the mixing rate. Others are controlled indirectly, but we want to try to match them. So how high of density the cells grow to, the product amount they make, the titer, and then how much byproduct or like metabolism byproduct or carbon dioxide accumulates. We don't directly control that, but it is controlled by how much the cells make and how much, when we're adding the air and oxygen to supply the cells with oxygen, that also can strip out some of the carbon dioxide. So we'll choose those gas flow rates and also to some extent the agitation rate to indirectly try to control the carbon dioxide accumulation. And then for how we choose the set points for some of the parameters, some of them are scale dependent. Some of them are just directly, some are independent, meaning if the temperature is 37 degrees at small scale, how we developed it, it's 37 degrees at the large scale, dissolved oxygen, pH set point, those are also, you just transfer them directly. Some of the other parameters though are scale dependent. So feed volumes, we scale linearly. So if we add 200 liters of feed to a 2000 liter, we'll add 1,000 liters to a 10,000 liter, straightforward. The gas flow and agitation rates are scaled non-linearly, and there are some equations that we use and to try to match either per volume flow rate, or sometimes we do it by the linear gas flow rate. Kind of depends on different sites do it a little bit differently, but then in terms of the agitation, depending on the configuration of the bioreactor, meaning what the mixing, we call it the impeller, looks like, and the shape of the tank and the volume, we try to match what we call power input, which is a complicated calculation based on the shape of the bioreactor and the mixer, and try to make that match. So the mixing rate in RPM won't be the same, but we're trying to get a more uniform power input to get the scales to perform similarly. For purification, again, operating conditions selected to try to get consistent performance at all scales. It doesn't mean every operating parameter is the same. So for solid removals, sometimes we'll try to not have, like, even if a site has a centrifuge, if we know we're going to eventually go to a site that doesn't, or vice versa, we'll design the process one way or the other. One consideration also for that step is if we are not doing a centrifuge step and just depth filter, you have to make sure that you don't have too high of pressure across that depth filter, because at that point you still have the cells in there, and if they rupture, they release enzymes that can reduce the product, meaning break the disulfide bonds and the product falls apart. At harvest, no good. So there are ways to make sure that you keep the pressure low, you can chill it, make sure the oxygen level stays high to prevent reduction. For liquid handling, we have to make sure that the pool, so at small scale, the various buffer volumes are one way, and then at large scales, sometimes the facility can't handle, the tank is just not big enough for the way that so you just have to keep in mind volumes of all these tanks, how long you can hold and have the buffer be stable or the product be stable. Also mixing speeds are a consideration for downstream. Chromatography, we keep constant across all the scales, the bed height, product concentration, residence time. and then scale up linearly the total load, volumetric flow rate, and cross-sectional area of the columns for the chromatography. And then for membrane filtration, we apply safety factors to make sure that at scale the operation is there's plenty of membrane surface area for the larger scale. Okay.
Gayle, we had a question come in. Is there a general rule of thumb for the amount of production and date of expiry to the proposed sample size or like the enrollment for the clinical trial? Sometimes we run across situations where the drug expires and there's a lot of waste, or the drug expiry is approaching and there's a lot of pressure to basically speed up enrollment.
Yeah, of course, in an ideal world, right, you only make the amount that you can use before it expires, but it is tricky, right? Because you don't always know enrollment, like you pointed out. You don't, sometimes if we're trying to go really fast to start these studies, we don't even know the study design, and we're just going to do a 2,000 liter because that's what we can do quickly, right? And so sometimes, yeah, then we do make too much, but yeah, we try not to overproduce as well. You don't, when you do know it, right, try to not, I mean, the just-in-time manufacturing concept, right, where make it when you know you need it, but it's a little hard for biologics because the lead times are pretty long for some of the raw materials, and even if you're manufacturing at a contract manufacturing site, their lead time just to get the capacity can be 2 years. So yeah, so I don't know if I exactly answered that question.
I mean, I think the answer is there isn't a specific, each team, each company has to kind of weigh the investment in how much material to make versus the length of the studies and strategize on that. There's no one size fits all answer.
Yeah.
But chances are you're not going to try to make enough to supply an entire large study right off the bat.
Yeah, especially like you don't want to make the phase 3 material when you haven't even passed phase 1, if you can avoid it, right? Because you might not pass phase 1 and then you wasted all the money. Yeah, it can be really challenging to figure that out, right? And to guess which programs you should pre-invest in and, you know, be ready for the study early versus which ones you wait until you're sure you need it, and then you're probably, the CMC is on critical path to starting the study, which isn't ideal either. Okay. Supply chain management. We talked a little bit about this, but the overall high-level view of the supply chain is the drug substance, drug product are manufactured. There's labeling not really shown on here. Primary packaging, secondary packaging, and then it goes, like I mentioned, to a storage depot, and then from there, I'm sorry, I think these are supposed to be the study sites. This is a mislabeled. And then the clinical team monitors how the study is going, make sure the inventory is there. For biologics, one of the challenges is cold storage, because most of our products are not stable at room temperature. And so it just adds another layer of complexity, making sure that you're tracking the temperature during transportation. We also have to do studies to support any potential shaking during transportation, any temperature excursions. We'll have that data ahead of time to make sure that if there is a temperature excursion, that we can show that product quality wasn't impacted. Yeah. Okay, I think that's actually all I have. I'll be back.
Let's be back at, let's make it 11:50, as Eric mentioned. All right, thank you, guys.
Okay.
Great, welcome back, everyone. It's 11:50 just now, and Eric, take it away.
All right, hopefully everybody can see the slides.
Yes.
Okay, so thanks everyone. As Dolo said, I work here at Gilead Sciences, and as Gayle's in our biologics group, I work in small molecules. So as you'll kind of see, there are a number of similar concepts in terms of critical quality attributes and things like that, but there are also pretty significant differences in terms of kind of how we actually control things, some of the challenges we run into in terms of scale up. And so rather than kind of a little bit more dry kind of nitty-gritty details of some of those concepts. This is structured a little bit more as a case study, like Gayle was saying. And so we're going to go through a little bit more around the formulation process development of a product called Harvoni, which is a combination of 2 different drugs for the treatment of hepatitis C. So for this, I've got a list of abbreviations in here. I'm not going to spend any time on them, but they're there for reference because there are a lot of acronyms throughout the presentation. And so if you need it, you can try to go back to reference. I have tried to do my best to capture them all. There may be a few that I missed, but hopefully they're all there. And then we'll go through this case study. So I'll go through a really quick kind of high-level overview of hepatitis C and what was available at the time when this was being in the early stages of discovery and development. The development of Lodiposphere, which was one specific compound that's an NS5A inhibitor, overcoming some of the challenges for that compound. So this is what's called a BCS class 2 compound. We'll talk just a little bit about that, but some of the challenges that that presents for us. And then finally kind of coming up with what's called a single tablet regimen, or STR, where we then combine that drug with another drug to give the simplest treatment possible for the patients. Okay, so again, abbreviations here for your reference. So like I said, I'm going to go through these next 5-6 slides pretty quickly because they're just kind of high-level information about the disease, which are kind of helpful to understand why we're working on this, some of the decisions that we make, but we're not going to go through all the nitty-gritty. So this is a viral infection of the liver. It was discovered in the late 80s. It's bloodborne, so it's transferred through use of needles, high-risk sexual behavior, things like that. Basically anywhere where you could get exposed to blood, like with HIV and other diseases like that. It can cause both acute and chronic hepatitis. Acute cases will basically clear on their own and are not a major concern. But about 85% of patients can't clear the virus on their own, and they'll end up developing chronic hepatitis C infections, which can result in cirrhosis, hepatocellular cancer, ends up causing the need for liver transplants, as well as being a major liver-related death contributor worldwide. So in 2013, it was a global health problem. It was estimated about 3% of the world's population, so somewhere around 170 million people were infected with the virus. And then from 94 to 2015, the annual death rate had quadrupled over that time. So ultimately, it was estimated that it was causing around 350,000 deaths a year. About 27% of cases of cirrhosis in the world were attributed to HCV infection, and about 25% of hepatocellular carcinoma cases were also attributed to these infections. Now, one of the difficulties of this virus was there are several different what are called genotypes, so several different genetic variants of this. So generally broken down into 6 categories. So genotype 1, 2, 3, 4, 5, and 6. And these have different distributions around the world. So you see that a lot of the so North America, South America, Europe, a large portion of Asia are all predominantly genotype 1. And then you get some of the areas of Africa, which are predominantly genotype 4, and some others. Now, these are not the only genotypes in these regions. These are just the predominant genotype in that region. And so basically, the hepatitis C treatments had evolved over the years. So for a long time, the treatment was inadequate. So the standard of care available until 2011 was a combination of subcutaneous pegylated interferon and oral ribavirin tablets that were administered. Now, what you'll see here is this started back in 1990. They had initially used just interferon was injected. Now this required basically, I think it was daily injections for 24 to 48 weeks, and essentially would make you feel like you had the flu for that time. And these only gave you a 10% chance of being cured by the disease. So really the goal here is to cure, not just to treat the symptoms of the disease. Now, in 1998, they had found that if they added ribavirin onto that treatment, so now an oral combination, they could now get those cure rates up to about 25% of the people that took the therapy. But the important aspect that's not captured here is not everyone was eligible for the therapy. There were a lot of people that wouldn't even go through the treatment because the side effects were so extreme, they were too sick to even take the treatment itself. So this was only the cure rates for those who actually were able to take the treatment. And then as of 2001, they had now changed interferon to utilize pegylated interferon. This was not really a change in efficacy. It wasn't really a reduction in side effects. This was really just to reduce the number of injections that were required. So by pegylating the interferon, you could increase in the circulating half-life. And so now you could go to maybe injection once a week instead of every day. But you still had all the same nasty side effects and other things. But now they've gotten to the point of having about 45% cure, mostly through increased compliance, things like that with the therapy, because you don't have to get so many injections all the time. All right. So eventually then, in 2011, we had found that we could add protease inhibitors, or PIs, to this regimen. And again, we're still treating for about 24 to 48 weeks, but they could get these up to about 96% cure rates. But you still have all the same issues with side effects, eligibility for enrollment that are associated with the interferon and all the nasty side effects. Ribavirin also had its own set of bad side effects and issues. So, all right. So at this point now, a new drug was becoming available, which was Sovaldi, which had one active ingredient, which was sofosbuvir. And this could be added to either the PEG or the PEG-RIBA. You didn't need the protease inhibitor. This could now achieve either 90% cure rates for genotypes 1 and 4, or genotypes 2 and 3, you could achieve about 67%, and you actually didn't need the pegylated interferon, which helped quite a bit. So sofosbuvir was a potent and selective inhibitor of what's called the non-structural protein 5b, so NS5b, which was just one of the proteins involved with the viral replication process. And it was effective against genotypes 1, 2, 3, and 4. So genotype 5 and 6 were not quite as effective against. It could be administered as a combination with other drugs, and it was administered as 400 mg once a day via oral route. Okay. So at this point, we're trying to see what else could we come up that we could add in with sofosbuvir or other drugs to then get rid of the interferon with the ribavirin and make a more palatable and kind of easier to use and less side effect prone therapy for patients. So Gayle touched upon the idea of a target product profile, or a TPP. So what is it that we are looking for out of the product that we're trying to develop? So in this case, we'd like to have once daily dosing. So we don't want to have people taking this 2 times a day, 3 times a day. So a lot of the protease inhibitors that they were using required taking those about 3 times a day, and many times it was 3 or 4 tablets each time. So a very high pill burden or tablet burden for the patients to take along with the injections. We'd like to have it orally administered, so we don't want to have any injections if we can. We'd like it to have it be an immediate release tablet. Now, what this is just, we don't want to have something where we need to develop an extended release that's going to slowly release that drug over time. It's just simpler to develop if we can do that. So we'd like it to be immediate release, where we can just release all the drug at once, it's absorbed in the body, and the clearance of the compounds are slow enough that you only have to dose it once a day. We'd like to have all of those agents contained within a single tablet. So this is that single tablet regimen idea. You can have everything that you need in that one tablet. You just take it once a day. This is a carryover. This was something that was really revolutionary for HIV treatments. It used to be that you would have to take a handful of tablets, like what's kind of shown on the left, a handful of tablets. For HIV, you also had injections. You have this complicated regimen to adhere to. And if you can get it to where patients only have to take 1 tablet once a day, it makes it much easier for them to be compliant and it much more palatable for them to go through. So we'd like to minimize drug-drug interactions. This is not necessarily specifically a formulation aspect. There are some aspects that are relevant to formulation, but not all of them. We'll touch upon the one that is formulation relevant, but there are other drug-drug interactions that are designed into the drug substance itself. We'd like to minimize any effects of food. So we'd like to not have to restrict if patients take the drug with or without food. And then also have a robust intra and inter-patient performance of the therapy. Okay. So we want to develop this STR containing sofosbuvir. So we've got sofosbuvir on the left. So this is the chemical structure. As we discussed, it's an NS5B inhibitor. And now we want to have there's a typo there, just NS5B, not NSA5B. On the right-hand side is then we want to have a second agent that we can combine with sofosbuvir in this single tablet regimen. And so one of the things we were working on was NS5A inhibitors. So inhibitors of a different mechanism of viral replication. Now, in this instance, we're talking specifically about targeting genotype 1. There were subsequent efforts to treat all genotypes. We're not going to talk about that today, but that was after this therapy was developed. We need to have sufficient potency and metabolic stability to achieve a low dose. So we can't have too high of a dose or we won't be able to fit anything into one single tablet. We need to have a half-life that's compatible with once daily dosing so that we won't have to use an extended release formulation where we slow the release because of fast clearance or short half-lives. And we also need to have a suitable drug interaction profile for combination with other HCV antivirals and complementary mechanisms. So what this means is it needs to not have a drug-drug interaction with sofosbuvir that we're trying to administer with it, and it needs to have a complementary mechanism. So they can't have the same mechanism of action. We need to have diverse mechanisms of actions. And this is mostly to prevent or minimize the risk of developing resistance to the drug by treating multiple mechanisms of action. Okay. So Gayle touched upon this, kind of this idea on the left-hand side here of preclinical. We go through lots and lots and lots of compounds. So for this specific project, between about January of 2009 and December of 2009, so basically over about a year, there was a lot of work invested in trying to find a potential drug substance. In the end, Lodiposphere, which is an NS5A inhibitor, was discovered as being highly potent with a long half-life that was going to be suitable for once daily dosing. And so that's shown on the right here, the chemical structure for Lodiposphere, or what's also known as GS5881. So a lot of times in clinical development, we don't have a name for the compound yet, so it'll have some sort of a designation. For this one, it was GS5885. And then eventually we come up with the generic name, which was Lodiposphere. So we've got the potency, estimated human metabolic clearance, as well as this idea of BCS class 2. So what this means is that the compound is considered to be highly permeable, but have low solubility. So the drug permeates well across, or should permeate well across, the gastrointestinal membrane, but it has solubility limitations. So it may not dissolve well enough to get enough of that across the GI barrier to get into the plasma and have a sufficient exposure. So if we can get enough dissolved, if we can increase that solubility, we'll have a better shot of achieving efficacious exposures. And then this log P just kind of has it's basically a measure of hydrophobicity. So how much does the compound prefer kind of a hydrophobic versus a hydrophilic or water environment? And so this 3.8 is it's moderate, it's fairly hydrophobic, that it likes kind of oily types situations more so than the aqueous environment, which is part of what contributes to the poorer solubility. So one of the important properties of molecules that we have to factor in is solubility, and this comes back in part to concepts around ionization and pKa. So Lodiposphere is a poorly soluble compound, and it is a weak base. So it has two different ionizable moieties within the molecule. There is a pKa for the benzimidazole, which is shown in the blue here, which is a pKa of 4, and then the imidazole has a pKa of 5. Now, what does this mean practically? So basically, when it is at more neutral pHs, it's unionized. As it gets more and more acidic, like what's in the stomach, it will start to ionize, and that will actually increase the solubility of the compound. So the biopharmaceutical performance is dissolution limited. Again, this gets back into the solubility. So a drug that's classified as poorly soluble will generally have a dissolution rate limitation for that bioavailability, because if it doesn't dissolve fast enough, it's going to transit through the GI tract before you can get enough of it absorbed. So if it doesn't dissolve fast enough, you're not going to get enough absorbed. So that's why we talk about dissolution limited. You may have low or nonlinear bioavailability. So when you give what's called a conventional solid dosage form, we'll talk a little about what does conventional mean versus what is non-conventional. But when you give this type of a formulation, they'll have these compounds will have a tendency to exhibit low bioavailability, but also it's likely not going to be linear. So as you give higher doses, you may see it cap out, or you may see that we give twice the dose, but we didn't see twice the increase in the exposure of the compound. You may see a lot of variability between patients or repeated dosing within the same patient. You're going to see potentially pH-dependent solubility. So like with this compound, like I said, under acidic conditions, it's much more soluble than under more neutral conditions that are present in the intestines. And so when you're giving things like an acid-reducing agent, such as omeprazole, things like that, you will potentially see an impact on the bioavailability of the compound. Because now the stomach pH is not nearly as acidic, it won't dissolve as much, and now you may see a much lower dissolution and thus a much lower exposure in vivo. And then also the potential for significant food effects. The presence of food causes release of bile salts, which may improve the solubility. And so you may actually see an improvement in exposures when it's administered with food. And so here's just a graphical representation of that solubility I was talking about. So under acidic conditions, you see a much higher solubility relative to the more neutral conditions that are present in the intestines. Now, additionally, there are a couple of different media that we use that you may hear talked about, which are generally referred to as FASF and FESF. So FASF is fasted state simulated intestinal fluid. So this is a media that we use that basically simulates the intestinal contents under a fasting condition. So you will have some bile salts present, and this essentially kind of replicates that pH and bile salt concentration. FESF, on the other hand, represents that same thing under a fed state. So you'll have a lot more bile salts present in it, and the pH is a little different because of the gastric process that happens during digestion. And so really, when we're looking at this, if you see under a fasting condition or a normal stomach pH, you'll see good solubility, but then you see about 100-fold drop in solubility for the compound in the intestinal contents. Now, this drop could significantly influence the bioavailability of the compound, because as the contents move from the stomach to the intestinal tract where it's going to get absorbed, the solubility is going to drop significantly, and so you may not be able to get good enough absorption. Now, if you gave it with food, you may see another 10-fold improvement over that fasted state intestinal solubility for the fed state intestinal solubility. And so this is where then we would expect we may actually see an improvement in exposure if we were to administer this to a fed patient. Okay. So these are all kind of informing us on the risks, and then the studies, we will talk to clinical pharmacology and other clinical colleagues to identify when we should be doing food effect studies or acid-reducing agent studies, things like this within the clinical study to understand the clinical risks. But we can also look at this within a dog to also have an idea of what sort of a risk might be present. And so this is now looking at bioavailability in a dog from a conventional formulation where we look at the penagastrin, the blue. Penagastrin is just used to normalize the stomach pH of a dog to similar to a human. The pH of dogs' stomachs tends to be very variable in a resting state. And so by administering penagastrin, we normalize that to be more consistent with a human fasted state. And then famotidine is to represent basically the administration of an acid-reducing agent within a human. And so what we see is a pretty significant difference in the left-hand side around the absolute, you see the actual plasma concentrations over time are significantly lower when the dogs were treated with famotidine before giving the compound. And on the right-hand side, you also see that numerically, where you see basically about a 7-fold drop in the percent F for the penagastrin or kind of normal human state versus the famotidine or ARA state. So basically, we had found this compound. It was potent. It had low predicted clearance. It had sufficient potency to achieve a low dose. It had a long half-life. It had no significant adverse effects noted within a 14-day tox study. But the issue we've encountered is that there's a food effect, potential positive food effect, and a potential acid-reducing agent or ARA drug-drug interaction. And so we'd like to try to fix these if we can. So how can we go about trying to overcome this solubility dissolution limited bioavailability for this compound? So we're always looking at biopharmaceutical performance, manufacturability, something that we can actually manufacture and eventually manufacture at appropriate scales and commercialize, as well as in vitro dissolution, which is one of our critical quality attributes we're using to kind of understand the performance of the compound and how it's likely going to dissolve within the stomach, as well as then the drug product stability. So there are a variety of dosage forms that we can look at. So liquid-filled capsules. So you've probably seen these as over-the-counter, like a lot of cough and cold medications come as these kind of soft gelatin that have a liquid inside of them. You can also have a hard gelatin that's also filled with a liquid. You can have solid dosage forms. So some of you may have encountered with other companies, you do a powder and capsule. So there's various ways of doing it. Basically, just take the powder and put it in a capsule, or immediate release tablets. Now, within that category, we have dry granulation and wet granulation. Now, we're not going to go in, we don't have time today to go into what is dry granulation, what is wet granulation. But at a very high level, dry granulation is basically you take powder, you shove it together under high forces to compact it into something, and then break it into particles again. But those particles are larger. Why do we do that? Think of powdered sugar. A lot of times that's what our drug substance is like. It's very fine. It doesn't flow very well. We need it to flow well on equipment at high speeds to make tablets and things at a high rate. And so by doing this process, we can take that powdered sugar and turn it into something more like sand or granulated sugar, where it will flow much better. And that just improves the processability for us. So that's dry granulation. Wet granulation does the same thing, but through the use of water. And you can also potentially add surfactants as part of that. You can add the surfactant in the water to help improve wettability dissolution of the drug. And then we also will look at the state, kind of the form of the drug substance. So in this case, we had a couple of different forms to work with. There was an amorphous form, which is very disorganized, generally high solubility, but also high energy and at risk to crystallization. And then a crystalline tartaric salt. So we crystallize tartaric acid with the compound to make a salt. Now, there are advantages and disadvantages to each of these. We looked at taking that amorphous material or the crystalline tartaric acid salt. If amorphous, we're going to focus on the top here as a non-precipitating solution. So if you take an idealized, it's dissolved in a vehicle, when it's administered to the dogs, to humans, it doesn't precipitate. So an idealized, solubilized drug, you see a bioavailability percent F of 53%. So this is generally the highest that we could probably hope to achieve. We probably cannot ever do any better than this with any solid oral dosage form that we develop, a tablet, things like that. Now, we're going to skip over the rest of those. You see that some of the other forms for the amorphous material, the bioavailability weren't as good. And then we look at the crystalline tartaric acid salt for an immediate release tablet. And we see that generally, even a couple of different doses, we see pretty good bioavailability. It essentially matches the non-precipitating solution. So great. We've got a solid oral dosage form using a salt that's easy to make. It's a conventional process, and we can basically achieve the bioavailabilities that a non-precipitating solution, the best that we could hope to achieve, was able to achieve. So how do we manufacture this? So we take that drug substance, that API, we mix it in with a bunch of intergranular excipients, a bunch of excipients in ingredients. We take it through a milling process, basically just to distribute that. We're not actually changing particle sizes. We're just helping to distribute that and remove lumps that might be present in it. We then add in some additional intergranular excipients. We put through that through a roller compactor, it's called. That's how we do the dry granulation process. Basically, the powder goes through a couple of big rollers to make a ribbon. We then break that ribbon up into small chunks again. We collect that, we add in some additional excipients. So now this is what we call extra granular, because we have granules and we're adding material outside of that as excipients. We'll take that final material, we'll put it on a tablet press to make tablets, collect those, typically put those through a film coating process to get coated tablets. There's a variety of reasons that we make coat tablets. Part of it's aesthetics, can help with swallowability, but also sometimes compounds are sensitive to light, and so the addition of that coating may also help to protect the compound from light. And so in the end, we end up with these tablets, and then we'll subsequently package them into bottles, blisters, whatever we need to do, and distribute them to the clinic. So this so-called conventional tablet contains crystalline salt of the liposphere, and it unfortunately ended up giving a limited exposure within the clinic. So what we see is an increase in the AUC as we increase doses up to about 100 milligrams, and then above 100 milligrams, we saw no further increases in the AUC. This may or may not be a problem. In this case, it was, because the efficacious level we hadn't been able to achieve. So this can't really proceed in the clinic any farther. Additionally, along with this, we had collected food effect data. And what we see is that in individuals, this is actually human data, in fasted individuals, and then in fed, we see about half the exposure in people that were in a fed state. So this non-conventional formulation approach is what we're going to have to go to. So what does that mean? But this is one of the ways that we will go about trying to improve the biopharmaceutical performance. There's different ways. One is to make a salt, which we already did. We already had this tartaric acid salt that we had used, and it wasn't enough. Another alternative is to develop a prodrug, so chemically modify the drug substance to improve solubility, permeability, fix whatever that issue is. The issue there is it needs to be cleaved as part of that process. It might be cleaved in the gastrointestinal border to basically release the active compound, which is absorbed. It may be that that prodrug gets absorbed and has to get cleaved in the liver or in systemic circulation in order to release the active substance of the compound. But it is chemically a different substance. In this case, we weren't going to go after that. That wasn't an option that was on the table. We could also look at adding acid or base modifiers to the formulation, so try to make the environment more acidic or basic while the tablet is dissolving. You could look at particle size reduction. So can we go and make the particles of APIs smaller to increase the surface area to make it dissolve faster? Or we can also look at incorporating wetting agents, so surfactants, things like that, to help make it easier for the drug to get wetted by the API, by the water, and also to just dissolve into the water. So these are all kind of conventional approaches, and these have been tried, and they didn't really work for the liposphere. So we needed to do additional work. So one is to go after solubilized formulation. So we talked about like liquid-filled capsules. That's another option. In this case, it was not a viable option because we need to eventually combine with sofosbuvir. So it wasn't even evaluated because it would not work for that application based on our TPP. And so the option we were left with was something called amorphous solid dispersions, where we basically take and make an amorphous material, but it is dispersed within typically a polymer matrix to help stabilize it. And it can also help wettability and other performance aspects. And so this amorphous solid dispersion is what we had gone with, and it gives a dissolution and solubility enhancement. And these can improve the drug exposure and bioavailability by increasing the solubility as well as the dissolution rate of the drug. So amorphous solid dispersions can improve this by increasing solubility and dissolution rate. So this method is basically you take this active ingredient, like we're saying, an inert carrier, and typically it's a polymer, and you can either melt them together or you can dissolve them in a solvent and use some sort of a solvent or a solvent evaporation. And so these are kind of the overall options here. It's basically a drug substance and then some sort of a matrix, and you can either use a technique called spray drying, where you're going to use a solvent, or melt extrusion, which is the melting option. So within spray drying, this molecular dispersion occurs in solution. You take and dissolve them all, and then you'll spray that into basically a fine mist, drive off the solvent, and you're left with the API and that polymer in that matrix as a solid that you can use. Conversely, for melt extrusion, that happens, there is no solvent or any sort of a carrier. It is simply the drug and the polymer or other additives that are heated, melted together under force and mixing, and then basically forced out and extruded into typically a rod that can be cut, sometimes for implants. So a lot of the implants that are used for birth control are processed through this method, but you may actually just chop that rod into a section that's actually used as implanted directly. In this sort of oral application, we will take that and further process it into a powder. So we'll take that and grind that up even more into a powder that we can use to put into a tablet and things like that. Okay. So what are the benefits of this? So we can create an amorphous material, which inherently generally has better solubility. It is a higher energy state. It'll give you this solubility enhancement. It'll generally improve exposure and bioavailability by increasing that solubility and the dissolution rate of the drug. We can engineer those particles. So not so important for this application, but if anybody knows or remembers the inhalable insulin, that is an aspect of important particle engineering. It's done through spray drying, and then by controlling the formulation, the processing controls, you can control the particle morphology to make particles that are basically easier to get into the air and get into the lungs. It's a continuous and scalable process. If we need to make more, generally we can just run the equipment for longer. It's a little easier sometimes for process scale up if we just need to make smaller increases in the scale of manufacturing. And it's generally easy to make, and the material is fairly easy to process. And actually, what this is getting into is if there were an issue, when we're spraying it into this mist, if there was a problem and it drips, we don't get good atomization, you can potentially just take all that material, redissolve it, and put it back in the spray dryer again. So it's not necessarily wasted. If I make tablets that are wrong, I can't really do anything. I can't recover the material. But with this spray dry dispersion, you can collect it, redissolve it, and put it back through the equipment again. What are some of the challenges? One of the big challenges is physical instability. So this is a high energy state. If at any point you start to get crystallization of the drug again, it is going to be very prone to recrystallize, and then your solubility is no good anymore. There's actually a really famous example of this sort of issue. It's not specifically from spray dry dispersions, but if anybody is familiar with ritonavir and its history, this was a drug that was launched, and then at one point, it was for HIV, at one point actually had to be pulled from the market and reformulated because a new crystalline form basically started crystallizing out of the formulation. And it caused basically to not be on the market for a couple of years and had to be relaunched. Additionally, this material is typically very hygroscopic. What does that mean? It takes up moisture very readily from the air. Now, why is this a problem? That water, that moisture in the material can reduce what's called the glass transition temperature. Basically, it makes the material more mobile and increases the likelihood and the potential for it to crystallize. Additionally, if you're trying to process this material, making sure that you get the right dose can be challenging, because if it's increasing and taking up 10% of its weight in water, as you're trying to weigh it out and process it, the doses that you end up providing in the formulation in the final dosage form are not going to be right. So that's why hygroscopicity can be a real issue with these sorts of materials. So development of this, so basically in developing this process, we have to consider what is the right form of the drug to bring in. So should we start with the amorphous material? Should we start with the salt? Or in this instance, there was actually a solvate, so the drug with acetone crystallized with it, that was also available. And so in the end, that acetone solvate was what was chosen for this process. We need to choose the carrier. What's that polymer that we're going to use? So there are many options. The two that are shown here are hypermellose, or HPMC, or hydroxypropylmethylcellulose, as well as copovidone, or the kind of one of the brand names is Colidone VA64. Now these have some different physical chemical properties, but in the end, copovidone was what was selected for this as being the best candidate for making the spray dry dispersion with liposphere. And then we finally also have to choose the solvent. So there are a variety of solvents available. Ethanol, methanol, DCM, or dichloromethane were all evaluated for this. Now, how are we considering what solvents to use? Because there are a lot of other solvents out there available. It needs to have good enough solubility. The compound is soluble enough in the drug to reach adequate concentrations to make this process viable. But also, it has to have a boiling point that is low enough that is reasonable within processing. So if you take something that takes a lot of energy to boil it off, you're not going to be able to process it because you're not going to be able to get rid of that solvent during that drying process. So it has to have a low enough boiling point to remove it. Generally, water is about as high a boiling point as we would use. Typically, they are lower. Okay, so in the end, ethanol was what was selected for this. Now, you may question, DCM's on there. Could you really use DCM for something that's going to go into humans? Yes, because we have to test and show that the residual levels of solvent after processing are low enough to be safe. So that's why things like DCM can be used, or methanol can be used, because we will have to test to make sure that the residual solvents are low enough to be safe for the patient. Okay, so we've done all this. We've selected the components that we're going to use, and then we have to evaluate the solubility, the stability, the in vitro and in vivo performance, and then ultimately develop the actual process. So we ended up choosing a 1-to-1 mixture of the drug to the polymer, and then going through and optimizing the actual spray drying process, the temperatures that are involved, and other parameters during the processing. And then eventually we get this LDV, or liposphere spray dry dispersion, or SDD. Okay, so it's amorphous and it's hygroscopic. So it's amorphous. We can confirm this by different techniques. One is powder X-ray diffraction, which is shown on the left. So since a lot of you probably are not used to looking at this sort of data, what we would be looking for is, if this was crystalline, if it had order, you would see peaks within this plot. Instead, what we see is a very broad, we usually say amorphous halo. There's essentially no major features within this. You can kind of see two lumps in the middle there, but those are not really significant. Those are not the sort of sharp peaks that we look for crystal materials. And if you see towards the left-hand side, right towards the y-axis, there is a little bit of a shoulder that comes up. Again, that's not crystal material. That has to do with, it's kind of stray X-ray signals that are close to where we're actually getting the X-ray source. So that's also kind of an artifact of the experiment. So it's good. It's amorphous, so this should be improving our solubility as well as our dissolution rate. Glass transition temperature, I mentioned before. So what is this? What does it matter? Glass transition temperatures are related to the mobility. As you take an amorphous material and you warm it up, you will get an increase in mobility, and it will be softer and start to flow once you get past a certain temperature. So this glass transition temperature is a critical temperature within that. If you are below it, you're in what's called a glassy state, which has a much reduced mobility, molecular mobility, and so you're less likely to have enough mobility to induce or get crystallization. When you get above that glass transition temperature, it's what's called a rubbery state, and it is much more mobile, and so you're at a much higher risk for crystallization. One of the examples that I've heard people talk about, now this is not crystallization, but in the concept of glass transition temperature, think of a piece of bubble gum. And if you're in the wintertime and you put it outside and it's cold enough, maybe not in the Bay Area, but if you're somewhere where it gets colder and you put it outside and it freezes and it gets really hard and brittle, that is essentially the glassy state. If you take and put it back in your pocket or something and it warms up, it becomes much more soft and much more malleable. That is basically above the glass transition temperature. Okay. So we would like to be below it, and we would like to be quite a bit below that temperature if we can. And the good thing is here, it was quite high. So you'll see in this plot, basically it was 140 degrees Celsius. So it is quite high, and it's less likely that it's going to crystallize. And then finally, this idea of hygroscopicity, we talked about how much water it takes up. Taking up the water increases the likelihood of crystallization. And so when it gets exposed to higher humidities, it's going to take up that water, and then you have that higher likelihood of crystallization. And so what this plot in the bottom is showing you is basically how much water, the increase in weight of the sample as you expose it to higher and higher humidities. And so if you can read it here, basically what this is saying is from 0% RH, so effectively bone dry, all the way up to a 90% RH, so pretty humid, you get about a 12% increase in the weight of the sample. So it's taking about 12% in water by weight, which is fairly significant. It's not necessarily turning into a liquid, but it is a high risk for crystallization. Now, part of what we do is we can put controls in place during manufacturing to limit this happening. So there are ways you can manage this during the manufacturing process to reduce risks. So the good thing is we take this solid dispersion and we expose it to different environments conditions. Now, this is not specific to spray-dried dispersions. You could do this for any drug substance, but in this case, we're looking at this amorphous dispersion. And what we see is that as we expose it to different temperatures and humidities, it remains stable. It remains amorphous in this case. Now, this is FT Raman. So you might look at it and say, well, there's peaks here. Why is this? Because this is using FT Raman or Fourier transform Raman as opposed to X-ray. So the important thing here is we don't see any changes in the spectrum, not the actual presence or absence of peaks. And so what this is telling us is if we store this at 25 degrees Celsius, 60% RH, closed, so it's protected from the environment, but we store it in that condition, which is a normal ICH guidance condition, or at 4075, which is an accelerated condition, this remains amorphous. So this is a good sign that this material is pretty robust and usable and doesn't end up showing any crystallization or phase transitions. Additionally, some additional work was done. So there's a 50 degrees Celsius, 92% RH, which is really aggressive. And the material was actually exposed to that condition. So it's very challenging. That remained amorphous. And also you see that chemically there was no change. So it remains 50% drug, which was the target. And you don't see any change in the total degradation products or impurities, which is the last column here. And then also with subsequent conditions as well. These all basically are showing that this is chemically stable as well as physically stable that we saw on the previous slide. So this is all looking good. We've got a material that's amorphous, the solution rate should be better, should increase performance, and it's now chemically and physically stable under a variety of different conditions. So you'll see here on the right just kind of a picture of this material. So this kind of shrunken raisin characteristics, and this is because of just the spray drying process ends up inducing this particle morphology. So we've now got a solid dispersion that's suitable for use in tablets. It's amorphous, it has a high TG, so it should be stable, and what we've seen it is stable. It should have a low propensity to crystallize. It's hygroscopic, so we're going to have to be careful about our handling and packaging configurations, but it's manageable. It's physically stable under open conditions by a couple of different techniques. And then it's also chemically stable for up to 6 months under accelerated conditions. So this is all looking really good. So now what we've got to figure out is, how do we actually get this into an actual product that we want to enhance exposure? We want to eliminate the DDI with acid-reducing agents. We want to reduce the food effect, and we want to reduce that variability between patients. So a new tablet was developed using the solid dispersion in order to overcome these issues. And so we take that STD, we mix it, again, same sort of process in terms of mixing it with these excipients, milling it, mixing it again, roller compaction, tableting, coating, and in the end we get this tablet. And so we've also shown this a generic composition here. It's got about 30% of the tablet contains this solid dispersion. About 64% is various fillers that just help to take up space to make a tablet big enough for you to be able to handle. And then 5% of what's called a disintegrant. So this just helps the tablet to break apart and come apart in the stomach to release the drug. And then also a little bit of a lubricant. Lubricant's important to minimize the sticking to the surfaces of the equipment. These will tend to stick to metals and things. So by adding just a little bit of a lubricant, and generally this is like a solid, basically surfactant essentially is usually what it is that's added, as opposed to not just your dish soap. That's not the type of lubricant that or like petroleum jelly or something like that. That's not the type of lubricant we're using. So, and then in the end, this ended up, it was a 600 milligram tablet, which is relatively large, but not nearly as large as your multivitamin tablet that you usually take. Okay, so how did this perform? So the plasma exposure of the tablets was similar to a non-precipitating solution in these famotidine pretreated dogs. So if you look, we've got the non-precipitating solution on the left. It had about 30% bioavailability at this dose. The conventional tablets were okay, but then you take the STD and now, well, and actually I guess they're okay. They're okay in the penagastrin treated dogs. They were not very good in the famotidine treated dogs. But now when you look at the spray-dried dispersion tablets, we actually get a bit of an improvement in the penagastrin relative to the conventional tablets. And then we also see an improvement for the famotidine relative to the non-precipitating solution. Okay, so we're essentially matching what the non-precipitating solution can do. So I guess basically this STD is doing about as good as we could ever hope for a solid oral dosage form. So this got taken to the clinic. So here now in comparison to that original tablet data from humans, we now see that as we get up to these about 90 milligram dose, we now get an efficacious exposure. So we've been able to improve the exposure in patients to where we should have an efficacious effect. And also when we look at the food effect, we now see essentially no difference in the exposures using the spray-dried dispersion in the fasted and fed states. Okay. So Finally, then the patient-to-patient variability. This is then, so the left and the right are the same data. They've just removed one of the outlier patients. But basically what this is showing is that we were able to reduce the variability between patients from the exposures. So you will see, you did occasionally get one patient that was maybe higher, but on average, you tend to get a higher exposure from the spread dispersion relative to what you see for the conventional tablets using the salt that was originally used. The one challenge was the spread dispersion did not overcome the drug-drug interaction with acid-reducing agents. And so when lidopisphere was given alone, you see this AUC here. When it was given with omeprazole as the acid-reducing agent, you still saw about a 50% decrease in the exposures. So it cannot just be used with any acid-reducing agent in any regimen. However, I'm not going to get all the details. You can then start to look at modifications. So can you use famotidine? Can you put a gap? Can you allow a gap of 8 hours between the administration of the ARA and this therapy? So you may be able to find certain regimens that would allow some ARAs, but at least in this case, we couldn't blanket allow all ARAs in use with the lidosphere. So the clinical trials for this STD showed improvement over that conventional tablet formulation. We had improved dissolution rate. We had increased bioavailability and improved linearity of the formulation. We were able to reduce the patient-to-patient variability, and we were able to eliminate the negative food effect. The one thing that we couldn't do was eliminate the ARA effects. We can find some certain situations to be able to allow the use of some ARAs. We just can't allow all ARAs under all instances. So now we've gotten to where we can improve and kind of get ideal performance out of the lidosphere. Now, how do we get to a single tablet regimen? So we've got the sofosbuvir. It's got its mechanism, this NS5B inhibitor. It has a dose of 400 milligrams. It is what's called a BCS class 3 compound. So its issue is not solubility, its issue is permeability. We need to make sure that the release of this compound is not hindered by the presence of the lidosphere, because if we slow that release, it may alter the exposures because of permeation challenges, not the actual solubility of the compound. It's crystalline itself. It's not hygroscopic. It's physically and chemically stable. And in the end, the single agent tablet, the tablet that contained just sofosbuvir, so that's Sovaldi, was 1,200 milligrams in total size. So 400 milligrams plus all the extra materials to get to 1,200 milligrams. So now we want to take and combine that with the lidosphere, which is an NS5A inhibitor. So we have a complementary mechanism. There's a dose of 90 milligrams, but we need to use 180 milligrams of the solid dispersion in the formulation. It's a BCS class 2 compound, so it has solubility challenges, not permeability. It's amorphous, it's hygroscopic, it's practically and chemically and physically and chemically stable. And that STD tablet itself was about 600 milligrams in total size. So we want to get to a single tablet. We would like to have it be less than 1,200 milligrams in total size if we can. It needs to contain 400 milligrams of soft and then another 180 milligrams of the solid dispersion. So right off the bat, we've got 580 milligrams right there of things that we have to have present within the formulation. It needs to have comparable pharmacokinetic performance to each of the single agent tablets. It needs to be physically and chemically stable. It needs to have a commercially scalable and commercially robust manufacturing process. And it needs to be able to be stored at room temperature. So the team had looked at a variety of different manufacturing processes in order to try to make this single tablet regimen containing both of these agents. So one is what's called a monolayer co-granulated tablet. So what does that mean? Everything is contained within a single kind of portion. So monolayer versus like bilayer. If you're aware of things like Mucinex, if you've ever looked at it, there's actually 2 different colors, or you even see this with some of like the laundry detergents where they'll have like different layers, different colors. Those are made by the same compression process that we use for tableting, but those layers are different layers within it. So same idea with a tablet. You can have different layers, And so you could separate actives between us. Monolayer means it is all the same thing. There is no stratification across the formulation. So it's all one thing. And then when we do that granulation process, we are putting both drugs in together. So they're in pretty intimate contact with one another. That's going to be the most efficient process. We just got one process flow. Everything's kind of in together. It has a high bioequivalence risk. So if the dissolution of each of them influence one another, they're in close contact. So that might be an issue. And there's high AP interaction. So if there's a chemical interaction, that can also cause stability issues. So there are a lot of risks with doing this, even though it's preferred because it's simpler. Another option is to keep them all one monolithic tablet. But when we make that granulation, when we go through that roller compaction process, separate the two drugs into different granulations. So now, even though they're going to be in contact, they're a little more isolated from one another. They'll be in separate granules that are next to one another instead of being in intimate contact. So it requires two different blending processes, but it has a lower bioequivalence risk than the co-granulated, and it has moderate API interaction. Now, the last is bilayer, which we talked about, where they're actually present in two separate layers within the tablet. This is going to be even more complex of a manufacturing process, but it has the lowest risk of bioequivalence. So the best chance that it's going to perform the same as the single agents, because we essentially can probably use the single agent formulations for each of the layers, and it's going to minimize the API interaction. But it's our least preferable because it's the most complicated one to develop. Okay. So we look at all these, we look at chemical and physical stability, we do in vitro dissolution testing, we look at dog PK performance, and then try to choose which one is going to be the best option for the clinic and for long-term manufacturability. And so this is now looking at dissolution, so how quickly the drug dissolves from these different tablets within a media. This is not necessarily intended to mimic the stomach or mimic the intestines. This is potentially just a media that is basically the performance of the product is sensitive in. And so what we see is the sophosphere on the top. Generally, all of these perform similar to one another and similar to the single agent, which is shown in the black. So we see pretty quick release and dissolution of the drug. And then liposphere also shows similar performance between these. You do see some subtle differences, but generally it's all completely dissolved within about 30 minutes. Additionally, we look at bioavailability, and they also performed similar across these different options for both of the agents in the dogs. So in the end, these were also chemically and physically compatible. So when we take that spray-dried dispersion and just mix it directly with sophosphere, so there's no other excipients present here, we just take and mix those, we compact them together into a mixture, put those on stability. That then basically showed no issues around chemical stability. We don't see any growth in impurities and degradants of sophosphere or liposphere in these. So they're all chemical. We don't have to worry about separating them into different layers to improve the stability because the two compounds seem to be chemically compatible with one another. They were also physically stable. So we didn't see any new crystallization of the liposphere due to the presence of this crystalline sophosphere. So in the end, we were able to select a 1,000 milligram monolayer tablet for the STR in the clinical studies. This is just the composition. So it's got 40% sophosphere by weight. It's got basically 18% of the spray-dried dispersion. It's got 16.5% of lactose, 8% MCC, 2.5% of cross-carmylose, 1% silicon dioxide, 0.25% magnesium stearate. And then once we've granulated all of that together, then it has some additional microcrystalline cellulose or MCC, cross-carmylose sodium, and magnesium stearate. So that results in the 1,000 milligram tablet. And then we coat that with a product called Opidry 2 that's orange, and we target to add about 3% weight to the tablet, is kind of how we control to get adequate coverage. And water is used in that process, but it's removed. So there's really no additional water in the final product. This is just the overall process. Again, it's similar to the ones we've talked about: mixing, milling, roller compaction for dry granulation, compress the tablets, coat them, and then you end up with, in this case, you can see here, these diamond-shaped tablets. These are physically stable for 24 weeks in the absence of any sort of a desiccant to help keep them drier within the packaging. The tablets were exposed to high moisture, were physically and chemically stable. So even if they do get exposed to humidity and take up water, they're still chemically and physically stable. We also have to look at them in the actual packaging configuration. So that was stable. And ultimately, we were able to support a shelf life of 2 years if they're stored below 30 degrees Celsius. So basically at room temperature. And this was using that monolayer co-granulated process. This has been taken to the clinic. So we have to do a relative bioavailability study to show that the single agents and the co-formulated product perform similarly. And so when we take the 2 single agents and administer them versus also taking the fixed dose combination or single tablet regimen. So distinction between fixed dose combination and single tablet regimen, they can be the same thing. Fixed dose combination just means that you have 2 or more agents in a fixed dose in that dosage form. Single tablet regimen is a fixed dose combination where everything is within a single tablet. You could have a fixed dose combination where you need to take 2 tablets, 3 tablets. It's not a single tablet. So an STR is an FDZ. It's just a subset where everything is contained within that single tablet. Okay. And so basically you see that for both of these agents, basically comparable exposures in the clinic. So this was all good. So in the end, this combination tablet was developed in less than 2 years. So the Lodiposphere itself, the preclinical phase discovery was about a year. IND was filed basically that next year. Phase 1 was started that same year. Phase 2 was about a year and a half later. Sofosbuvir was launched as a commercial product as a single agent in combination with interferon and rotavirin in 2013. But then in parallel, we were developing the combination of the 2. And so those phase 1, the relative bioavailability study of the co-formulated product was in 2012, so kind of during the phase 2 studies. We were able to execute phase 2 at the same time. And then in 2013, executed the phase 3. And then finally the NDA was in 2014 and it was on the market in 2014. So basically that fixed dose combination from starting it until having it on the market was about 2 years. So I have a single tablet regimen that contains 90 milligrams of Lodiposphere and 400 milligrams of Sofosbuvir. The tablet was only 1 gram, so it's still smaller than multivitamin. It's not small, but it's still smaller than a normal multivitamin tablet, so it's manageable for pretty much all patients. It's developed as a co-granulated monolayer tablet using an amorphous solid dispersion of Lodiposphere in combination with Sofosbuvir. So kind of the simplest process we could have from manufacturing of the tablets, and a pretty scalable process for the spray dried dispersion. Ultimately, this was developed and scaled up to a tablet process of about 450 kilos per batch. We were able to develop a physically and chemically stable tablet that contains amorphous spray dried dispersion. All the GMP batches of good manufacturing practices, batches of Sofosbuvir and Lodiposphere film-coated tablets met the acceptance and release criteria, so they were acceptable for administration in the clinic to the humans. We were able to demonstrate bioequivalence of that combination tablet to the single agent tablets. We were able to achieve something that could be administered once a day orally, and it can be administered with or without food, and it can be administered with famotidine or omeprazole. There were just some restrictions around how that actually administration was done in some of the timing. So Harvoni, this combination was approved in 2014 and was able to improve HCV treatment outcomes significantly. When it was rolled out, Harvoni delivered cure rates in the mid-90s in as little as 8 to 12 weeks of treatment with limited adverse reactions. I'm not going to read through these, but there was just a couple of kind of testimonials from some of the different patients and care providers. I know I have met patients. I didn't work on Harvoni. I worked on the subsequent product, which was pan-genotypic, which covered all genotypes. And I know I've met patients and how appreciative they were to be able to get their life back from these therapies or with these therapies. this is just, I'm not going to go through this in detail. This was just kind of the actual labeling around the treatments for the different genotypes. But we were able to make significant advances for the patients in terms of quality of life and ease of treatment and ease of use. So with that, I know it was fast and I apologize, but I wanted to make sure to give us time as well to try to answer questions afterwards. So I will open it back up for questions.
Thank you so much, Eric. What an excellent presentation. I'll never look at a tablet the same ever again after this. Really fantastic, life-saving treatment. Any questions? So do you want to just take the questions from Slido and just do it yourself, or do you want me to read them?
Sure, I can do that. That's fine. So I'll just start with, it looks like the kind of where we started at. So are animal tox studies ever combined with bioavailability studies? Not really. Generally, you wouldn't want to, because most tox studies are going to be multi-dose studies. And so it's not going to be basically worth your investment to try to capture that with a bioavailability study. Yeah, I can't think of an instance where you would really want to combine them. And you're usually going to be doing bioavailability studies to establish the formulation and the doses that are going to get used in the actual tox studies. Those don't even necessarily match with studies that we would do to support efficacy studies, or that we're doing to support our human formulation dosage development. Because I'm working on developing a tablet, we're not going to administer a tablet as part of the tox study. So at least for most small molecules, they're not going to necessarily overlap. Are the dogs in the fed state to understand affecting famotidine? Typically, when you're looking at things like famotidine, you would be doing that fasted. And then there is a separate set of studies you would do to look at the fed effect in the dogs, where you would then take and look at feeding the dogs, but it wouldn't be in conjunction with the famotidine. And there are standardized meals, just like in the clinic, where you'll have standardized high-fat and low-fat meals for dog studies. Different companies have different strategies around, you just give them wet dog food. Do you actually use the basic composition of the high-fat meals, things like that would be used in the clinic. And there's different strategies depending on what you're looking at as to how you might do that. But it is a consideration. So. Why was an immediate release tablet %F higher than the non-precipitating solution of an amorphous drug? I don't remember which slide that was on. This one? So I think this is with respect to, so a conventional tablet is immediate release, why this number is higher than this. And so this is where you don't actually have the comparator. So this is penagastrin and this is famotidine. So the comparator here is actually this column. And so it's 9%. So it's actually lower. So if I missed something around that, please reply and let me know. But I believe this was the slide, and that's the reasoning, is that the treatments are different for this column versus this column. So it's this versus this, or this. And you do actually see here the conventional versus the spray-dried dispersion. Even the penagastrin, you do see here then the improvement in performance from the spray-dried dispersion. But we don't have the non-precipitating solution here for comparison. Regarding dissolution method for crystalline drug, how fast the dissolution profile was, and what was the timing for developing discriminating dissolution data you could remember? Okay, so trying to digest the first part. So the last part around when to develop a discriminating method depends on whether you are talking about for quality control purposes or for formulation development purposes. We will typically try to pick something that is going to be discriminating as early as possible, but it's for development purposes. So it may not be a method that results in full release, or it may not be as robust as what a QC method might need. In terms of developing a kind of discriminating method, we would say, for QC purposes, for kind of regulatory submission. That is typically more kind of, it'll depend from company to company, but typically more kind of in phase 2. It might be in preparation for phase 2. It might be during phase 2 in preparation for phase 3, but you're going to need it in place before phase 3 because you're going to need that for your essentially commercial product. And so you want to be testing your phase 3 supplies with that sort of a method. In terms of the first part, I'm a little unclear on exactly what the question is. If the question is why crystalline drug is so fast, again, it's for sofosbuvir. It is very fast because the solubility is very good for the compound. Solubility is not the limiting factor. And so actually one of the things that you can see in looking at those profiles here, you see that even within about 5 minutes, the sofosbuvir is essentially completely dissolved. So essentially, as soon as the tablet disintegrates, the drug immediately dissolves. For lidiposphere, it is a slower process. Now, if you were to look at this in water, you would not get all the liposphere dissolved. If you looked at it in FASF, it would not all dissolve. This method is more of a QC method. This is not representing what is happening in vivo. This is a method that is more geared towards really distinguishing and differentiating formulations. So this is more of a QC method, not an in vivo representative, if that makes sense. Why did liposphere show negative food effect even after having high solubility in FESF versus the FASF? It's probably in part a pH issue as well. The pH of the stomach will increase in the fed state, and so it's probably a mixture of several different effects. It's not completely as simple as just FASF versus FESF solubility, but differences in FASF and FESF solubility can give you an idea of risk. But in the end, in all cases, ARA, DDIs, food effect, you're going to have to test it in the clinic and either show that there is or isn't an effect. You can't just solely rely upon the in vitro solubility measurements. Okay. Regarding dissolution method for amorphous drug, did you follow if it is crystallizing over time during dissolution study, and how do you prove it? Yeah, so this is an important issue. It turns out in this case, crystallization is really not a problem. The compound really just does not like to crystallize, but I have worked on another project where it was very difficult to develop a dissolution method because the compound did have a lot of propensity to crystallize during the dissolution method. And so it is something where most dissolution testing, you are looking for full dissolution. It is fully dissolved. And so if you're seeing complete dissolution, 100% release, it's all dissolved, there's nothing crystalline present. If, however, you're not seeing full release, that is where something is still present as a solid, and you start looking to find out, is it because it just didn't fully dissolve, or is it because it crystallized out of the media? And so you may have to use a variety of different techniques. So hopefully that answers the question. There's a lot of different nuances to that we can talk through, but I'm not sure we want to go into it right now. So I'm happy to stick around and talk with whoever it is afterwards as well if you have more questions around that. What's the requirement for developing quantitative X-ray for amorphous drug to quantitate residual crystalline? So this is a good one. There is no specific regulatory guidance on when it's needed, but generally, if you are trying to develop an amorphous compound, they will likely be requesting that you test it in some way. Now, do you need a quantitative method? It will partly depend on what your dissolution method can detect. If your dissolution method is sensitive enough to crystallization, that may be sufficient. There may be other instances where maybe you need an additional method that is triggered by dissolution testing. There are others where you may just have to test for crystallization. And in part, it depends on company policy as well and company strategy around how they approach doing it. Some companies are very risk averse and they may just add a quantitative method regardless. Some, for a variety of reasons, would prefer to only trigger it in certain instances when needed or to investigate an issue. But it's always a negotiation with the agency based on the situation in terms of the risks. Because in the end, it is ensuring consistent performance for the patient. And how are you going to do that? And that's the key, and that's what you've got to answer. So why is crystallization important? So yeah, I kind of flew over this. So as an example, and this is not truly differences in crystallization, but one of the things that's used to illustrate this is if you think of carbon, you can have diamond, you can have graphite. They have very, very different properties. Diamond, pretty much one of the hardest substances on Earth. Graphite, you write with all the time on a piece of paper. They're both carbon. And so you can have different crystalline states, different crystalline packing of the molecules. And that's essentially kind of what graphite and diamond represent. They can have very different properties in terms of solubility and things like that. Amorphous is just a higher energy system. So it's even more soluble. It's even less stable than some of those other crystalline forms. Now, most companies will opt to develop the most stable crystalline form of a molecule. And what does that mean? Generally, it is the least soluble version of the molecule. So why don't we just choose other forms that are more soluble and less stable? Because they're less stable. There's a lot of risk. That's what happened with ritonavir that I mentioned before. The company had looked and looked and looked for different crystalline forms, and they couldn't find one. So they came up with a formulation, they launched it. Several years after the product had been launched on the market commercially, they by accident discovered a new, more stable crystalline form. It's less soluble. And so basically it wouldn't dissolve in dissolution tests. I mean, this is the dissolution method caught it. They took them a while to figure out what was going on and how to overcome it, but they caught it. And so in the end, they had to reformulate to overcome that one. That was actually a liquid-filled capsule, and they had to come up with a new way, a new formulation, a new process to actually get that new, less soluble form to dissolve into an acceptable formulation. But that's in the end, but why? I mean, crystallization, the more stable crystalline form is generally less soluble. And so if that's not what you're starting with, you have a lot of risk to performance impacts on the product. It can be done. So like this, there have been other amorphous solid dispersions that have developed on the market, but there's a lot of risks. And so you have to have the adequate controls in place. Have any companies tried to use 3D printing to make solid dosage forms? Yes, there is at least one product on the market. I can't tell you off the top of my head what product it is, but there are actually 3D printed tablets that are on the market. They are generally not for high volume products. So you will probably not, at least not now, see a blockbuster product that is a 3D printed dosage form, but they are out there. Glass resistant temperature for Colidone V64 is around 110. Why did the amorphous drug dispersion show a TG of 145? So yes, basically the drug substance has a significantly higher glass transition temperature, and that is why you essentially see, as you turned at the anti-plasticization effect of the drug. The glass transition temperature of mixtures are generally kind of a weighted average of the two components. Most of the time, drugs will have very low glass transition temperatures, and so the polymer is helping to increase the TG and make it more stable. This is actually an unusual situation, although I've now encountered it several times where the drug actually has a higher glass transition temperature and the polymer is actually lower. And so you end up with a lower TG for the mixture. But yes. How long did the development testing for the STD take and what cost? I mean, in part, we sort of captured this. It was about, well, from the STD beginning, it was maybe about 3 to 4 years from when the STD was first started to work on until we had commercialization. The cost, I would struggle to give you a number on that. It's expensive, but as with any product. Percent impurities are you okay with? Okay, so it looks like there's less than 1% in LVNC. So what's acceptable for levels of impurities and degradants? it's all a negotiation. It all depends on what the data is that you have around the toxicity and just process control. Is the agency going to let you have 10% of an impurity? Probably not. It might depend on what that impurity is, but it's probably unlikely. But what a safe level for an impurity or a degradant is established based on toxicity and risks. So you have to have some data to kind of back up and demonstrate that it should be safe. And there's a variety of ways that's done. We could probably do a whole lecture on considerations for impurities and different guidances that are out there around how to evaluate them and the risks. But generally, they're going to probably be less than 1%, but it depends. I have also seen cases where it is higher because it's a degradant and you just can't control it, but it's safe. So it's deemed as being acceptable to have 1.5, 2, 3, 4% for that product. So, with the introduction of STD, does it mean that compatibility only needs to be conducted between the API and the STD? No need for the two APIs themselves. And also the control of the polymorph of the API is not mandatory. So in terms of the API compatibility, there's no specific requirements from like a regulatory perspective. That's partly up to a company's basis in terms of how they do compatibility, assess compatibility risks. You could do the two APIs together. In this case, this data was around the one API and the solid dispersion from the other, in part because there was a concern around potential induction of crystallization of the liposphere in the presence of crystalline sofosbuvir. But it's up to a company. In the end, what matters is the final product is stable. That's what really matters. So there's a variety of ways you can get to that answer, but in the end, you have to show that the actual drug product that you're making is stable. In terms of control of the polymorph of the liposphere API, it just kind of depends on what the options are. In this case, there weren't really too many options for the incoming material, so there wasn't really much need to control it. But if there were different polymorphs, things like that might be relevant, you have to ask yourself, what's the potential implication? If I were to have the other polymorph, would it impact that manufacturing process? Would it mean that it won't dissolve in the solvent sufficiently for that process? So it's all just a matter of evaluating the attributes of the different materials and then deciding what controls might be needed. Any thoughts on why fasting improved drug bioavailability in humans with the conventional tablet? So it's not really that the fasting helped it, it's just that the presence of the food likely increased the pH of the stomach, just didn't dissolve as well. And so it's sort of like the ARA effect. It's just that the increased solubility because of the bile salts isn't helping you as much as you would hope. So it's really more of a pH effect there that's likely causing that reduction in bioavailability with the fed state. Given the variability of the dog stomach pH, why do dog studies, when they're also fed unfed media equivalents, do the dog study to also look at other organs? Generally, we're doing the dogs because that way you're getting additional information other than just the solubility impact. You're capturing some of that impact around permeation, things like that factor into the overall bioavailability, which is ultimately what we're trying to do. You can study in other animals. You could try to do in cyno monkeys. You can do other species. You can do mini pigs, things like that. But generally across the industries, dogs are the default model for small molecules for oral exposure. But yeah, we'll do as much as we can in vitro, but then sometimes we just have to go to the dog just to understand the likelihood before we go into the clinic. Why was IR tablet %F higher than a non-precipitating solution? I think maybe we already addressed this one. Slide 18. Slide numbers off, so. I think this entry like 68 versus 53. I think this is just a variability. It doesn't have the error bars here, but I believe that this is just higher because of variability. The variability can be fairly high in the dogs for this compound. So, And then are companies like Gilead and others turning to LNPs for drug development? Like right now, an LNP. So remind me, I'm blanking on the abbreviation.
Liquid nanoparticles, I'm guessing.
I mean, yeah,
they're-
and I was like trying. I mean, they've been around for quite a long time. It just depends on the product you're developing. I guess, Gayle, did you have?
Yeah, like I would say we don't have any right now in development, but we're always open to whatever the best modality is to get a treatment out there, right? So if that's the right way to do it for some mechanism of action, we're open to it.
And actually, I think possibly Amazon, I'm not sure the nanoparticles, but Amazon, I believe, uses this type of technology potentially. That's been around for a long time.
Yeah, I thought the ones I'm thinking of are like maybe putting like mRNA or something or some inside liquid nanoparticle to deliver to a certain location driven by some signal. But,
Maybe a question from me to both of you. How do startups think about investing in CMC early on versus as they move towards the clinic and get larger? What is the thinking around that for small molecules and for biotherapeutics?
You want to start?
I can start. That's fine. I mean, for small molecules, a lot of times for smaller companies, their main goal is to get through the initial phases of clinical development to show promise, many times to either license the compound out to a bigger company or sell it off. So sometimes their goal is to really grow and develop it and market it themselves, but many times it may not be. So a lot of times with those companies, their focus is really just minimal to get through the studies. So they may not be factoring in, oh, I don't care about acid-reducing agents. I don't care about food effect. I don't care about the long term, because I just need to get through these phases to get money from bigger investments for something like that. Whereas, say, a bigger company may have a lot more line of sight towards long-term development. Now, they may strategize it differently. They may still say, we're going to minimize investment to streamline early because there's a lower likelihood of success. But they're potentially going to be stuck with that compound for a lot longer than a smaller company is. So they may be paying attention to certain things that a small company might not be.
Thank you, Eric.
Yeah, I haven't actually worked at a startup, but I would say most of that applies for biologics too. And what I've seen is they don't have the capital to invest in the infrastructure, right? So everything has to be, it's like a small CMC group, like 1 or 2 people, and they oversee stuff going on at like a CDMO, contract development and manufacturing organization, where it's expensive to have them do it, but it's far more expensive to build the infrastructure for yourself for one molecule, right? To develop and to manufacture. So it like seems like I see a lot that they're just overseeing one of the CMOs that really does this a lot, and then eventually like they get bought, and then it becomes whoever buys them, it's on them to do all the other work that wasn't done, or bring it in-house.
I mean, as somebody who's worked on compounds that we brought in from smaller companies, I mean, it's usually a lot of the initial work is, okay, what are the gaps? What are the things that we don't have yet that are unknown? What are the risks that that's going to become a major issue, or just something that we're going to have to manage and overcome later? That's kind of your typical things you're looking at. And it's we understand, say, it's your focus is different, the priorities are different, and that's understandable. So
thank you.
We did have a couple more come in here now. So understand that IV and oral are main routes of drug administration, but do you have any words of wisdom for dermal administration or other innovative, ingenious methods under development? I mean, yeah, there's a variety of options. Those are the 2 primary ones. I mean, there's also rectal with suppositories, which is not as common anymore, but it's out there and gets done. There's inhalation. Each one of them poses their own challenges. And part of it, you really need to factor into the discovery of the drug. So if your goal is to develop a dermal application, you're going to want something that's going to be more lipophilic, it's going to permeate into that skin better than something that's just highly water soluble, what you might be looking for oral delivery. So part of that just factors into the discovery of the compound to begin with. All drugs are not going to work equivalently by different routes of administration. But then when it comes to the actual delivery, then say there's a whole number of other, say, respiratory delivery, say inhalation, and a whole set of things. And part of it, I like to factor in is there's a lot of things we have to factor in. What's the ultimate goal? There's been times where we need local delivery. Do we need systemic exposure? What is it that we're trying to achieve to understand how best to develop the compound? What type of a formulation might work best? If you want local lung concentrations, respiratory delivery might be your best bet. If it's a different issue, that's one of the issues that when they developed inhalable insulin was they wanted to get around injection, and it's going to get chewed up in the intestines, in the stomach. So, okay, well, we can get systemic exposures if we can get it absorbed through the lung, and we can bypass some of those other issues, or other options where you bypass first-pass metabolism, not having to go directly through the liver by not going orally. So there's a number of factors you have to kind of evaluate in terms of what's going to be the best way to try to deliver a drug, and will this drug work by one route versus another? I guess, Gayle, anything additional to add there?
No, not really.
And it is, I mean, IV and oral parenteral is the same either. Say IV versus subcutaneous versus intramuscular. There's different requirements in terms of allowable volumes and things like that. So it gets very complicated very quickly. Are there any highlights from today's pre-reading that we should be aware of? I guess, Gayle.
Can I just ask you a question, please? And what about when you are developing a drug to be delivered slowly with a implant in your body? Do you need to change something in the formulation, the concentration, and so on?
Yeah, so longer, say, in implantable devices or other kind of longer acting injectable products, it gets very complicated. Focus on the implant for a minute. There's different ways. So there are some that are non-degradable kind of matrices, that it is just the properties of the material that are slowing the kind of dissolution and release of the drug. Those will need to be surgically removed at the end of the time period. So it might be that it's implanted for a year, for 5 years, whatever it is. You need to have a way to visualize that, to remove it. So you usually have to have something to make it radio opaque, so that you can actually see it on an X-ray to define it. Because while you may insert it here in the forearm, you may find later on it's actually migrated over here to the chest. Like it has happened where those have migrated in the body, and you think that it's not possible, but you need to have a way to be able to find those to remove them later. There are others in terms of, say, just items that are injected that may solidify or otherwise modify to release the drug slowly over time. And they get very complicated. Yeah, there are a number of things to evaluate. It's still a very complicated and evolving field. So there's a lot of work going on with HHV right now around long-acting injectable. And it could be partly that it's through the properties of the compound, low solubility, low clearance. It could be part of the matrices. So like I was talking about the implants, or other components that do biodegrade, but they just do so very, very slowly to basically slow the release. There's a variety of approaches, and you kind of have to just figure out, for the disease you're going after, for the drugs you're working with, what's going to achieve the goals. And there's still a lot of questions in that area because it's still a new and evolving area. Anything to add, Gayle?
Yep.
And I guess the other aspect is, it is also can be a little challenging around the idea of impurities and degradants, because you're going to have something that's going to be in the body for a very long time. Is the compound going to degrade significantly into things that you don't want or need? The impurities that are present when you administer it, if you're do you have to treat those as those impurities are going to be released immediately, or that they will be released over the same duration? Because that may have implications on the specification for the product. If you have to treat it as it's released immediately for something that's supposed to be run for a year, you might have to control things to very, very low levels compared to treating it as if it's those impurities and degradants are going to be released at the same rate as the drug substances. So, and there's no clear answer from regulators. So, like I said, it's constantly evolving and moving target. So,
Okay, thank you.
Yep.
The pre-reading question, highlights? No, not necessarily. It was just kind of a supplement to the things that we did talk about in the biologics section, because I didn't really talk at all about setting specifications and how you choose the ranges that are acceptable. And there's a little bit on the early stage versus late stage in there too, but no, nothing that like specific that was needed there. How much of processes are done in-house versus at contractors for an organization like Gilead? I guess I could answer for the biology side. Trying to think of what kind of percent, but so for clinical manufacturing, that 2,000 liter scale that I was talking about and the pilot scale, we have our own internal manufacturing for drug substance. And so as many of the early stage programs as we can, we fit in there, which is for now, it's all of them. And then if for commercial manufacturing, we don't actually have our own manufacturing. So for a commercial product, we are using 100% contracted manufacturing. And then we also, not all of our products come into our pipeline directly from research. Some are through acquisitions, like Eric was mentioning. And so sometimes those are already being manufactured at a contract manufacturing organization. And in the interest of not making a change for the sake of it, we'll just keep manufacturing there. So we have quite a mix. I can't even think like what the exact percent is, but it's a good mix. So we end up, it's good. We learn the ways that various other external contract manufacturers manufacture as well, right? So you can make sure and design your process to fit anywhere.
And similar for small molecule, pretty much all of the development is done and or at least managed internally. So the aspect of development that's not always executed internally might be larger scale kind of process development optimization studies. As we're getting ready for commercial launch, we may do those at the contract site that we're going to manufacture at, but it's still kind of run and owned by internal development goals or groups. In terms of the actual kind of manufacturing, we have an internal drug substance manufacturing facility, as well as some capability here in-house at our site here, as well as drug product. We have a manufacturing facility in Ireland. Clinically, a lot of the manufacturing for the drug substance is done at our internal sites, and then external sites are used primarily as we get to larger scales for phase 3 and beyond. For drug product, it's a little different. Most of that manufacturing is done externally, both clinically and commercially. But we may have some commercial manufacturing at our internal site. Yeah,
I forgot to mention drug products for biologics. Yeah, most of our drug product has been external for now, but we do have one internal line as well.
Yeah, wonderful questions and fantastic session. Okay, maybe this is a wrap then. Thank you so much, Gayle and Eric. Fantastic presentation again. Thanks to the audience for being so engaged. We still have 22 people in the line waiting until the very last minute. Again, thank you for participating, and have a wonderful rest of the day and wonderful week. And for the rest of the audience, I'll see you back for the next session on Thursday.
Yeah.
Bye, everyone. Thank you.
Thanks, everyone.